Okay, welcome everyone. I'll ask you to take your seats. So it's fantastic to be together and be in the same room. The symposium this morning is scheduled to start at 8:30, but we invited everyone here for 8 o'clock. I don't think anyone checked the agenda because everyone showed up at 8. But imagine that, we get to spend some time and visit with each other for 30 minutes, things that we haven't done very much of over the last 3 years. Today's a very special day. We're here for the Don Weaver Symposium to take place, and it's really to honor the work and the impact that Dr. Donald Weaver has had here at UHN, at the Krembil Research Institute, at the Krembil Brain Institute, and really impact that is felt around the city, around the country, and around the world. And it was 10 years ago, almost to the day, that Don and his wife came to Toronto, and Don accepted the position of the research director of what was then the Toronto Western Research Institute. And I think it's very rewarding for us to think and look back over those 10 years and just how much has changed and how much impact Don has had here as a leader. When we recruited him, this was done in part through a valiant effort from many different people. Dr. Chris Page is here, who is the director at the time, and it was Chris's vision for what the Krembil could become, or what we know of the Krembil today. It was supported by the Krembil family and the Krembil Foundation, this wonderful new facility that we get to work in every day, and which has served as a stimulus for the recruitment and the development and the continued expansion of the Krembil Research Institute. But Don was a very unique recruit and has a number of unique qualities. I learned yesterday from Don there might be at least one other person out there with a similar background in the world, but there aren't many. And I think when you layer in a few of other Don's attributes, there isn't anyone like him. So Don's a neurologist, and for 10 years here, he had a clinic and saw patients every day, and also saw the scope and the scale of the problem that we're here to solve. And Don has dedicated his life to thinking about how to take care of these patients, and this has also driven his scientific interests to think about new solutions, new therapies, and new approaches that simply aren't available today. And we have many patients like this at UHN where the needs of those patients aren't being met the way we would like them to. And this serves as a stimulus for research across the entire organization, certainly at the Krembil Research Institute, the Krembil Brain Institute, and in Don's lab. And Don is also someone, I think, who has had a major impact not only on the science, but also on the culture. And we're going to get to hear today from many of the leaders at the Krembil Institute and how Don's impact has changed and influenced their own areas of research, but also the culture, the students, the collaboration, and the camaraderie that has occurred here over that last 10 years, something that's hard to quantify, but certainly has tremendous impact. And as all of you know, about a year or two ago, Don made the decision that he wanted to go back to the lab and dedicate all of his time to an area that he's particularly passionate about in areas of medicinal chemistry, and creating new molecules and new opportunities and new ideas for therapy. And so after his 10-year leadership role, where Don was placing the needs and interests of all researchers first, Don is excited to be back in the lab and spending 100% of his time back on solving those other big problems. So we're going to have a couple special announcements later on today, so please stay tuned for that. And we've got a terrific lineup of speakers here today. You're really in for a treat, and Don will be one of those at the end of today. But I want to get us started this morning, and to do that, I'm going to invite Dr. J.D. Baines, the new director of the Krembil Research Institute, up to the stage. And I think, Don, one of the best measures of success and impact is leaving the institution stronger and much stronger than when you came. And it was one of the reasons that we were so fortunate to be able to recruit the next leader here to this extremely strong and vibrant institute. So, Dr. Baines.
Hi, everyone. It's great to see a wonderful turnout this morning. It's my pleasure to be here today. This really special event. It's an event to celebrate and honor Don Weaver's achievements, contributions in shaping the Krembil Research Institute into the world-class research institute that it is today. So the story of Krembil is a story of innovation, it's a story of risk, it's a story of generosity. It's a story about trying to address the needs of the patient of today while planning for better outcomes for the patient of tomorrow. It's a story of visionary leaders who identified key needs for society and pushed to do things differently. Now, in 2023, we take for granted that clinicians and scientists work closely together. They work hand in hand to address some of the big challenges for human health. But in the 80s, which is when the seeds for the Krembil were first sowed, this wasn't necessarily the way things were done. In 1978, the Playfair Neuroscience Unit was established through a large gift from Mr. Stuart Playfair with the aim of developing a research center to study neurodegenerative disease, specifically Parkinson's disease, from which his wife suffered. Under the leadership of Peter Carlin, Charles Tatter, both of whom are still key members of our institute, neuroscience research flourished beyond Parkinson's disease. Peter reinforced the need to invest in research infrastructure, researchers, and clinicians. This primed the institute to be a leader in neuroscience research in Canada. In 1999, the institute was rebranded as the Toronto Western Research Institute, or TWRI. By 2004, under the leadership of Chris Wallace, research at TWRI expanded into the areas of arthritis and vision. This period also saw many important developments at the Institute, which included positioning it for a purpose-built research tower, made possible by a very generous donation from Bob and Linda Krembil, a large CFI opportunity, and additional philanthropy. That gave rise to this beautiful building that we're in today, the Krembil Discovery Tower, or KDT. So KDT opened its doors in 2013. About the same time, Don Weaver joined as director. On November 2015, the Institute was rebranded as the Krembil Research Institute in recognition of the Krembil family and the Krembil Foundation's cumulative support. At that time, it was over $80 million. From here, a number of new directions were launched, including the birth of 3 research institutes and formal recognition of allied health professionals and within the Institute. This 3 institutes in 1 model we have at the Krembil, facilitated by research and clinical co-directors in each institute, strives to bring together the clinical and research excellence in each of these areas. So the Krembil Brain Institute was led by Dr. Weaver and Dr. Gallaudet-Zadeh. The Schroeder Arthritis Institute is led by Drs. Mohit Kapoor and Rob Inman. And the Donald K. Johnson Eye Institute is led by Drs. Valerie Wallace and Efrel Mandelkorn. And you'll hear from one of the co-directors of each institute later this morning. And I'll leave it to them to share with you the focus and mission of each of their institutes. In addition to these 3 areas, we also have research programs that are outside of the institutes, which speaks to the breadth of the work going on at the Krembil. The Krembil has grown considerably from its original entity as the Playfair. Each year, the Krembil annual report captures our research excellence. And as I look through past reports, I'm delighted to see the increasing impact of the research conducted here, not only in metrics that we can measure, such as those on this slide, but also the impact we have on patients who who we're here to help. If I were to share with you all of the major discoveries made at the Krembil in the last decade, we'd probably be sitting here for most of the day, so I'll just mention a few. So Mike Timyansky, early in the 2010s, developed a drug that protects the brain from stroke damage. Those clinical trials are still ongoing. Mohit Kapoor discovered biomarkers related to spine osteoarthritis. Soumya Viswanathan had the first North American mesenchymal stem cell trial for treating knee osteoarthritis. Valerie Wallace discovered material exchange during photoreceptor transplantation. Michael Failings discovered novel neural circuits that restore breathing following spinal cord injury. And Dr. Pereira did the world's first robot-assisted brain aneurysm surgery. So these are just a smattering of the highlights of the Krembil Research Institute. Let me talk a little bit about Don and Don's tenure at Krembil. So during his tenure, Don focused on a number of initiatives, namely to grow the research capacity at the Institute, to support our trainees, who are the next generation of scientific leaders. And he did this through dedicated funding whenever possible. Don leveraged philanthropy to recruit 23 scientists during his 9-year tenure. He introduced a computational neuroscience hub. He established the Center for Medicinal Chemistry and Drug Discovery. He launched a number of grant programs like the Small Equipment Competition and a collaborative seed grant that brought basic and clinical research together. Don also did a lot for the trainees, provided resources for Postdoctoral Research Award, Krembil Brain Institute Postdoctoral Researcher Award, studentships in dementia research, and also collaborative trainee competitions to work with our colleagues at CAMH and the TANS. Don's mission, beyond the science, has been to make science and research more accessible and engaging for everyone and for the benefit of everyone. As he said in the past, science achieves little if it stays in the lab. In an effort to take Krembil discoveries outside the lab, he, along with the Krembil Admin Office and Krembil Public Affairs team, have run some very successful events that showcase our research to the public. Events, though, are not the only way to take science outside the lab. Don and his team took advantage of multiple communication channels, social media, annual reports, branded magazines that are a team effort with our UHN Foundation, Public Affairs, and Strategic Research Initiatives Development Department. And most recently, via a KBI-branded podcast entitled Your Complex Brain. I'd encourage you to check that out. It's really a fantastic podcast. So I think it's hard to sum up the impact that Dr. Don Weaver has had on the Krembil in just a few slides. He's built and fostered a diverse culture of research excellence across all levels, whether it's scientists, clinicians, trainees, or staff. I personally feel extremely privileged to be able to carry the baton from here and take Krembil to new heights. Thank you, Don. I'd like to ask Dr. Gallery-Zadeh, who's the clinical co-director of KBI, to come up.
Okay, thank you very much. I'm Gallery-Zadeh. I'm a neurosurgeon. And Don, this is really nice that we have the opportunity to celebrate you while you're present. I joked with you outside. And to really recognize you for the many contributions. I don't have slides, and I did think of what I'm going to say. Where's Helen? I apologize. I may have gone off the script that you instructed me to do, because I wanted to share with you how Don has impacted my career, and through 2 stories reflect on my interactions with him and how I learned from him. So about 10 years ago, Don is a neurologist, as you heard. I'm a neurosurgeon. He's cerebral, where I'm technical, and so he actually provides the brains for what we do on a day-to-day practice in our neurology, neuroscience world. So I work on acoustic schwannomas. They're difficult tumors to resect. They're sometimes very challenging. And some patients come to you and say that they take this drop that they buy from the farmer's market, and it works. And in fact, actually, anecdotally, it does work sometimes in some patients. You follow the graph and the tumor stays stable where it was growing, or on occasion it decreases in size. And if you Google it, you will see that there was attempts at the clinical trial to use this extract of propolis. So I mentioned this to Dr. Tatter. I don't know if he's here. He used to, in his prior life as a neurosurgeon, manage acoustic schwannomas. So he said to me, well, we have this really wonderful, smart medicinal chemist now. Why don't you speak to him and see what's in this drop? And maybe you could do some research on it in the lab. And I was desperate. I had just started as a clinician scientist. I would jump at any suggestion and idea, and I thought this may be my ticket to success. I emailed Don Weaver. He responds pretty much immediately, and I go to his office up in the big office upstairs on whatever floor I forget, and I explained to him this drop that people take, and the tumor gets smaller, and he just stares at me. And I think, well, he's liking the idea. I keep going. And he keeps staring at me. But with hindsight, I know that he was entertaining me because he actually went to the effort of calling in one of his colleagues to come and speak to me to see how we can find the active ingredient in this extract of propolis. I now know that it was preposterous of me to do this, and he entertained my idea because he really genuinely wanted to help me. And they did actually look at this drop that they themselves went and got from the farmer's market. Thank you very much, Don. And he emailed me back within like 2-3 months. I left the office thinking, he probably thinks I'm really not that smart. And how did I actually make it through medicine, let alone get employed at UHN as a clinician scientist? But I thought, well, we'll see what happens. I have the cell lines of the schwannomas. He'll tell me the drug. I'll treat them. There will be a response. I become famous. I find a drug. So about 3 months passes. I go to his office, and he says, we did the extraction, and there are, I don't know, many thousands of derivatives in it. 432 are active. And I said, oh, so which one of it is it? He said, well, you'll have to test all of them. So I'll tell you where that project ended. But just reflecting on what he did for me was really he entertained an idea that was rather outlandish, probably not scientifically sound, but he provided that support, and it meant a lot to me. So thank you, Don, for that interaction. Now roll forward a few years after that event. I, in the meantime, never interacted again with Don Weaver. I was asked and selected to be the co-director with Don. And I have to say that although at the time this role was fairly disputed, debated as to what the best phenotype would be, Don was nothing but supportive and worked with me very closely to introduce the Krembil Brain Institute to help eliminate some of the Krembil confusion because of the many contributions of the Krembil family to our institution. Keep turning this off. And so together we worked to creating the Krembil Brain Institute. And in the very first stages of me being in this role, he was extremely supportive. He never questioned my position. He never actually questioned any of the proposals or ideas, still continued to be outlandish. And we worked together to putting all of the documents together, to creating a strategic plan and strategy that we have a nice output for and continue to build on. And I think for me, that really encapsulates how Don provided what we refer to and recognize currently very important to support leaders as allyship. So thank you very much, Don. And I know that as a neurosurgeon, probably you don't think as highly of me as a neurologist, I have to say. But I do appreciate you entertaining all the wonderful and outlandish ideas that I had. But genuinely, I think you do embody allyship. And at no point did you really question any of my thoughts or suggestions or directions that we had to take together. Thank you very much. I don't see Helen in the room, but I apologize for going off script. However, I am to introduce Dr. Valerie Wallace next, who is the co-director of the Donald K. Johnson Eye Institute and is a unique scientist because she works on brain tumors, which is my area. I know her from that field, and then obviously eye pathologies. Thank you.
Good morning. It's a pleasure to be here today to FET Dr. Weaver. And my task is to tell you about how he helped evolve the what is now known as the Donald K. Johnson Eye Institute. And do I advance the slides? Yeah. We're on a mission to help restore vision to people who have intractable vision loss. So just to set the stage, over 5.5 million Canadians currently live with either blindness or severe visual impairment. A lot of these diseases are age-related, so obviously that number is going to grow over the next 20 years. And I have to do my plug about why it's relevant in terms of where these diseases happen. Yeah. And people go blind for a lot of reasons. Things that happen at the front of the eye, like corneal disease, cataracts, inflammatory diseases. There are autoimmune diseases of the eye, as well as diseases that affect the neural component of the eye. So that's this retinal tissue that lines the back of the eye. And I've spent most of my career reminding people that the retina is part of the brain. And some of the big causes of age-related blindness are macular degeneration, glaucoma, diabetic retinopathy. But even though this is rare, there are a number of patients with inherited retinal diseases. And because these neurons can't regenerate, this leads to intractable vision loss, massive unmet clinical need. So in 2013, when Dawn joined, vision research was a division of the Toronto Western Research Institute, and it was headed by Marty Steinbach. And there was a program of wet and dry lab research to look at disease mechanism, visual system development, because that was going to be important for any kind of regenerative therapy for the eye, and the psychophysics of vision. I mean, how do you actually see? How does the brain interpret what the retina sends it? As well as vision rehab, because actually that's feasible to do. And we also were in a unique position through philanthropy and somebody with a lot of foresight. We had a scholarship program for students that supported vision researchers across the city. So in 2016, after I arrived, the Donald K. Johnson Eye Institute was created, and currently I'm a co-director along with Ephraim Mendelkorn. And this was an opportunity to bridge that basic clinical divide by consolidating research, clinical care, and education. And our goal was to make advances in understanding disease and to translate that knowledge into therapies. And we've done a lot since 2016 to create a kind of harmonized and evolving structure where we increased the basic research program, and we expanded that from 3 to 6 investigators working in these various areas, including cutting-edge human stem cell modeling, which will be essential for understanding disease. Bridging that gap is a drive to recruit clinicians, scientists to kind of be that liaison between these worlds that seem to be siloed but shouldn't be, and looking at, again, disease mechanism and translation. And we've also established what is now becoming a citywide hub for evaluation of new therapeutics in animal models where we need specialized surgery and vision testing equipment, and building clinical capacity for clinical trials research in various areas, including low vision, retina, glaucoma, cornea, and inherited retinal disease, as well as vision rehab. So how did we get there? And really important, I could highlight what the group has done. They're funded, they're cohesive, they have shared resources. But Dawn was really, really instrumental in making this happen. This didn't happen out of nowhere. And a couple of ingredients that we really, really needed to do this. Well, first of all, is money. Over the last 10 years, we've attracted over $80 million in philanthropic support to make this happen, but it doesn't happen in a vacuum. It needs a lot of support because Don recognized that this was a strength at the Crimble and wanted to have it evolve and grow. And I'll just highlight two gifts that, again, Don was instrumental in helping us achieve. One was a gift, both of them were from Mr. Donald K. Johnson and his wife, Anna McCowan. In 2015, he donated $10 million for basic and clinical research, and that allowed us to recruit more researchers, and it formalized our clinical trials unit. Again, solidifying this ultimate goal to bridging that gap between basic and clinical. And in 2021, Don, again, was incredibly generous and donated $50 million in for basic and clinical research. And that is, again, allowing us to evolve our clinical trials unit. We're renovating space and expanding that operation. Again, recruitment, and we were able to recruit a clinician scientist. And again, a huge piece, if you really want to do this, you have to have some sustainability to the program. You have to have operating resources, and you have to be supporting academic activities. So another big influence that Don has had on even how we structured these gifts and how we are moving forward is that ultimately the end goal is, and this was Don's vision, he's a fierce advocate for the Crimble Research Institute, and his vision was to build research synergy and collaboration. No group is going to figure things out by themselves. He could very easily see the ties between arthritis, immunology, neurodegeneration, and retina. And a lot of times advances come from knowledge that's gained from outside your own area. And Don was adamant about fostering this. And that kind of vision influenced a lot of the leaders at the Crimble in how they built their programs. So first of all, he created a really solid governance framework with strong academic standards across the Crimble. And an administrative structure of excellent people with considerable corporate knowledge. He brought in resources and directed new fundraising and resource acquisition like CFIs to what would improve the greater good at the Crimble. So not just of the individual sub-institutes. So that took the form of cores and platforms. So when groups were lucky enough to apply for those resources, it was critically important that those resources be structured in a way that benefited and raised everybody's game. And a lot of Crimble-wide initiatives, Jaydeep mentioned these, but they're really important: studentships, collaborative seed grants. I see people collaborating who you never would have thought would have even been in the same room together. And again, I think that's where advances come, where people who never would have rubbed shoulders start to talk. And it's also his leadership style. It was very open, he consults, he's transparent, and there's strategic decision taking. So another thing about Don as a leader, and I think it's unusual, and I've definitely benefited, is he allows people to flourish. And when people do well, that actually makes him look good, and I think a lot of people would do well to remember that. But I think that's at the core of this. He wanted to make this a great place to do science, a great place to work, and I think he's achieved that. So on a personal level, he's incredibly approachable. Now, I won't say I won every argument I had with Don, but I did win some. He won some, but nobody walked away feeling bad. And he was very, very fair. He also was willing to provide really sage, non-science advice. I won't lie, a lot of us have had a lot of stuff in the last 10 years, not just scientific, but on the personal side. And it was always a good thing to be able to talk to Don. And finally, he was one of those people that I could share my childhood bad TV tropes with. And he got it, and he knew exactly what we were talking about when we mentioned the cone of silence, for example. So, Don, thank you. It's been a pleasure working with you. This place has definitely changed. It's better, and it's a very exciting place for all of us to work. So thank you. So it's my pleasure to introduce our next speaker, Dr. Sue Dillon. She is President and CEO of Biotherapeutics. Right now, her company is developing a novel pipeline of first-in-class siRNA molecules for orphan and genetic immune diseases, and I think we're all aware right now about siRNA, and it's amazing to see that deployed. But I was reading her bio, and it's kind of daunting. So she had a 16-year stint at Centocor and Janssen Pharmaceuticals. She led immunology R&D, and she achieved numerous regulatory approvals for antibody products to treat autoimmune disease, even some of which I'm familiar with, like Remicade, Simponi, Stelara, and Tremfya. She led multidisciplinary teams that went on to be recognized through awards, like the Prix Galien Award for Promacta and Stelara, first-in-class medicines. So she did her PhD in immunology at Thomas Jefferson University, and a postdoctoral fellowship in immunology at Duke University. She's had a lot of awards. I'm not going to list them all, but she was awarded a Distinguished Alumni Award from Thomas Jefferson. And I love this one. She was named one of the Fierce Women in Biotech by Fierce Biotech. She currently serves on the board of directors of Vendetta Biosciences as the vice chair of the board of trustees of the Wistar Institute in Philadelphia. We're so lucky to have you here today. I know that Dawn really wanted to have you here. I'm glad it worked out, and welcome.
Good morning, everybody. I hope you can see me above this podium. Thank you very much, Dawn, for inviting me. It's been great to meet a lot of you yesterday and today. I'm truly honored to be here, and I wanted to use the time to tell you about a few of the projects that I've worked on during my career, mostly in industry, and it all starts in drug discovery and research that eventually, sometimes, leads to truly miraculous drugs. And that is what has motivated me over all of these years. So we're going to talk about immunology, drug discovery, going from monoclonal antibodies all the way to silencing RNAs. And just give you a little brief snapshot here of some of the things that I have really had the privilege to work on and lead amazing teams of scientists, some chemists, a lot of biologists and immunologists and clinical scientists over the years, working on small molecule initially to develop a drug that had the unique property of being an agonist for a hematopoietic receptor, which was the first example, and it might still be the only example of such a molecule. That was developed for thrombocytopenia, and it treats all different types of thrombocytopenia. And then the two antibodies listed here that I'll spend most of the time talking about this morning, Stelara and Tremfya, were the work that I led at Janssen. And these are really amazing molecules, both because they do a phenomenal job for patients, but also because they unraveled the biology of the diseases that ultimately we found that we were able to treat. And then finally, I'll just give a little bit about what we're doing at my company, Arrow Biotherapeutics, looking at silencing RNA or small interfering RNAs as a new drug class to treat a variety of genetic diseases and immune-mediated diseases. So if we start with the story of Stelara. So Stelara is a human monoclonal antibody, and it recognizes a protein called P40. P40 is a subunit of both IL-23 and IL-12. And kind of the interesting story about this antibody is that when the research teams discovered it, it was actually discovered as a neutralizer of IL-12 because IL-23 had not been described yet. So we took this antibody into the clinic thinking that it was an anti-IL-12 antibody. It turned out that it was actually blocking both of these very important cytokines. A few years into the clinical studies, a scientist at a company that was then called DNAX in California described IL-23. And it described IL-23 as also having this P40 subunit. So we ran back in the lab and we discovered, lo and behold, what we had already started clinical trials with actually neutralized 2 cytokines, not 1. This turned out to be extremely fortuitous because, as I'll show you, the really bad actor in a number of immune-mediated diseases, as are listed here, is actually IL-23 and not IL-12. Years later, understanding the biology a bit better, we set out to discover and develop another antibody that was specific and is specific for IL-23. And it gets that specificity because this monoclonal antibody actually binds to the P19 subunit of IL-23, which is unique to the IL-23 cytokine. So both of these drugs work across many different immune-mediated diseases, and I'll just tell you a little bit first about the story of Stelara. That was really a miraculous discovery. And just a bit about psoriasis and psoriatic arthritis. I know there are many experts in the room, but very briefly, psoriasis is a disease of the skin, plaque psoriasis, and it can be relatively mild. It's extremely common. Up to about 2 to 3% of the entire population have some mild form of plaque psoriasis. But up to 30% of people have a moderate to severe disease. And as you can see in this patient shown on the left, this can cover literally almost the entire body, can cover any part of the body, very, very painful. And it is a systemic disease. It isn't just the skin lesions. It also has a lot of comorbidities that are very serious. About 1/3 of people that get psoriasis eventually develop psoriatic arthritis, which is a disease of the joints and the tendons, also a systemic disease that goes beyond those target organs and can be quite debilitating disease. So if we look at what's happening in the skin of a patient with psoriasis and has these awful plaques that can cover, again, a lot of the surface area of their body, what we see classically are these immune cells. And in this case, the immune cells are not good. They're not normally there. They're essentially have invaded the skin. And you can see in that middle panel there, the brown cells are T cells, T lymphocytes stained by CD3. And in the bottom panel are dendritic cells, stained by CD11C+. So these are the bad actors in psoriasis. They drive the disease. And what we would later discover with Stelara is that by treating and seeing the skin lesions literally disappear over time, so did these immune cells disappear from the skin. So just very briefly, this is Immunology 101, so I sort of apologize in advance if this is very basic for some of you, but I did want to highlight some of the cytokines or interleukins that are critical in driving psoriasis and many other related autoimmune diseases. The interesting thing about psoriasis, although the treatments are so amazing these days, is that nobody still understands what triggers this disease. There is definitely a genetic predisposition, but it is not a monogenic inherited disease. And if you have so-called the wrong genetics or the genetics that place you at risk, it doesn't mean you're going to get the disease. There needs to be some sort of environmental trigger or a series of triggers, which could be chemical, could be infection, that at some stage tips the balance and triggers the appearance of clinical disease, which is then a lifelong chronic disease, not curable so far. So initially, the innate immune system, shown there on the far left, kicks into gear, And the key thing that happens from all of these cellular interactions is that you get activated dendritic cells. If you remember those dendritic cells that are shown as being very prevalent in the skin. The dendritic cells are kind of like the traffic cop in immunology. They direct how the rest of the response is going to go, and they basically allow the adaptive immune response to kick into play. And here there are two different classes of T lymphocytes called Th1, shown at the top there, or Th17 cells. And these are generally believed to be bad players in many different immune diseases. Th1 cells are driven by that cytokine IL-12, if you remember, that we originally were going after, whereas Th17 cells are driven by IL-23. What happens once Th17 cells are activated is they secrete IL-17, thus the name. And IL-17 has a very direct effect on keratinocytes, keratinocytes being the most prevalent cell in the skin, and they cause keratinocytes to hyperproliferate, and that's what gives rise to these plaques. So I will come back to this diagram later. You don't have to memorize it, but there are obviously many, many cytokines that are involved in driving what the clinical disease ultimately looks like. So we took this antibody, Stelara, into the clinic, and I'll show you the results of one of the phase 3 trials that we conducted. And this is just an excerpt of one of the groups, but the first thing to look at here is simply the patient on the left. So obviously a picture tells 1,000 words, and this is a person who had a very high so-called PASI score that just stands for Psoriasis Area and Severity Index. It's exactly that. It's a measure, a composite endpoint of how severe psoriasis is, how much of the body surface area is affected. So this person, pre-treatment at baseline, had a score of 15.8. And here at the 52-week point of the study, you can see that they had a remarkable response in terms of clearance of these skin lesions. If you do the math, their score now of a PASI score of 3, it's about a 75% improvement. So this means that while the lesions are not completely cleared, because that would be a PASI 100, they are substantially improved from prior to the trial. So that measure of a so-called PASI 75, which means a 75% improvement, is what is shown in the data on the right, which represents hundreds of patients in each group. On the red the red open circles are the placebo patients up until 12 weeks, where they cross over to Stelara. That's the hatched lines. And the two groups that received Stelara at the beginning of the study are shown in the solid symbols, both light blue and purple. That just shows 2 different doses. And the y-axis there is just looking at the proportion of responders. So a couple of really interesting things from this study. The first is that it takes a long time. So you don't see a full response until almost 3 months. You see almost nothing if you look at 2 weeks or 4 weeks. 8 weeks, it starts to increase, and by 12 weeks, it's at its maximum, and basically it's not going to get better than that. So you're, in this case, at a 75% response. And as you can see here, it lasts out through week 40, and actually these studies have now been carried out for well over 5 years, and remarkably, most people do not lose response once they have a response. So the other thing that's really interesting about Stelara is about how infrequently it's dosed. So those arrows at the bottom of the graph there are showing when the drug is actually administered. It's a subcutaneous administration in a pen, like an EpiPen. And you can see that people are dosed at week 0 and week 4, and then every administration after week 4 is quarterly, so every 12 weeks. And again, this every 12-week regimen has been continued now for, some people have been on Stelara for well over 10 years at this point. And as I mentioned, usually the response is quite stable. So a really, really remarkable result with a drug that neutralized both IL-12 and 23, even though we came to understand that IL-23 was the key bad actor in this disease. So if we look at a couple of other endpoints, because we're in the Neuroscience Institute, I just wanted to show you that in addition to clearing the skin lesions, Stelara had a very meaningful impact on the dermatology quality of life scores, which are shown on the left, and on other scores, hospital anxiety and depression scores, which are quite remarkable improvements versus the baseline. So Stelara really kicked off a revolution in treating this disease, psoriasis, as well as psoriatic arthritis and other diseases, and provided a tremendous improvement. If you look there at about 70% of people, as I showed on the last slide, are reaching this PASI 75 response. The first thing that was approved in psoriasis years ago, Enbrel, which was around 2004, achieved about a 50% response. So that's very significant that 20% additional people who are treated will be able to reach that level of response. And now we have gone on to test this hypothesis that IL-23 is the bad actor and developed an even more potent antibody that is very selective for IL-23, which is called Tremfya, which actually reaches a 90% PASI 75 response and also treats very many patients to get to a PASI 100, so complete clearance of their psoriasis, which is really a remarkable result. So if we just come back to this very complicated graph here that's in all the textbooks, it was the clinical research with these phenomenal antibodies that provided tools to carefully dissect these mediators in the immune system that ultimately proved what is the most critical cytokine, not only in psoriasis, but also as it has followed in psoriatic arthritis, in Crohn's disease, in ulcerative colitis, and the list may still be growing. So I'll just end this part of the talk by saying that the journey is still actively ongoing. We have these remarkable classes of cytokine inhibitor drugs, and we've now 4 different classes of drugs, and I should say for each of these targets, there's probably 5, 6, 7 different drugs that have all been approved with very similar results. Very nice efficacy and very good safety in psoriasis and psoriatic arthritis. However, it gets more complicated when you go to other diseases. Interestingly, in inflammatory bowel diseases, which include Crohn's and ulcerative colitis, the IL-23 drugs work, the anti-TNF drugs work, but IL-17 drugs do not work. And in fact, they tend to, in some patients, even exacerbate that disease. So it tells you that although there are many shared pathways, there is not complete fidelity at all across these different diseases. Rheumatoid arthritis is still a puzzle in that the anti-TNFs, which were introduced very early for rheumatoid arthritis, are again a mainstay of treatment, work quite effectively, and yet none of these newer drugs work. We tested them, and they do not work at all. Tells you again, the etiology is completely different in rheumatoid arthritis, for example, than psoriatic arthritis. And I've only listed here multiple sclerosis, sarcoidosis, where none of these drugs work, but it goes on. Lupus is in that category, Sjogren's syndrome. There's a very long list of immune diseases that are not addressed by these drugs. And that is the focus of the work that we're doing now at my company called Arrow, because we need to get at new molecular targets that cannot be addressed with antibodies because they're not a cytokine or a surface protein. And in many cases, they can't even be addressed with a small molecule. So that takes us to Arrow. So Arrow is a company that I co-founded about 5 years ago. And our whole journey here is around creating a brand new class of genetic medicines that are receptor targeted, meaning that we have a way to carry these genetic drugs to the right tissue where the disease-related gene resides. And this is necessary for the potency and also for the specificity of these new class of drugs. And everybody knows, of course, about RNA drugs. We have vaccines that are miracles with COVID-19 that are made from messenger RNA. There are other types of RNA drugs that are being developed now. There's literally dozens of different types, something new almost every week. What we are focused on at Arrow are a class of drugs called small interfering RNAs, or siRNAs. And as you can see on the left there are already several drugs approved that are siRNA drugs, but these are very recent. The very, very first approval was in 2018, and several of these were only approved in the last year or two. So as I mentioned, these drugs are remarkable because you can target literally any gene. They are sequence specific. However, they don't have great drug-like properties. They're very poor uptake into cells. Not like chemical small molecules. They generally do not get into cells at all or very inefficiently. And they're very labile in the blood. So if you inject one of these siRNAs, the half-life, the circulating half-life is going to be less than an hour. So, immediately that tells you that you have a lot of problems to overcome. And these challenges have largely been overcome for delivering these siRNA drugs to the liver. But that's the only place that so far this has been achieved. Some of these types of drugs are also administered locally. For example, there's an intrathecal administration of related types of RNA drugs, but nobody yet has been successful in giving a simple sub-Q or IV injection and then having the drug go where it needs to go and get into the cells that you want it to get into. So that is our whole approach at Arrow, is to address a broad range of diseases with these siRNA drugs. So just a little bit more about how siRNA drugs work. The chemistries, this is all nucleic acid chemistries, are very well defined at this point. The mechanism is very well understood. And because they work by sequence-specific hybridization to a messenger RNA, they are extremely specific for the gene of interest and can be engineered to be quite potent. The other remarkable thing about silencing RNAs is that although it's very hard to get them into a cell, once they are in a cell, they have a very, very long durability inside that cell, and therefore the pharmacodynamic half-life, in other words, the time of seeing the effect of gene knockdown by an siRNA in humans is very, very long. Some of these drugs that have been approved that I showed on the prior slide are only administered once a quarter or even once every 6 months. So that provides a great advantage in terms of convenience for patients. However, I have told you about the limitations of these drugs. They don't get into cells very well, and they're cleared very quickly. So our technology allows us to overcome those difficulties. We have a small protein that we are working with, and that is proprietary to our company, called a centurin. I've showed a picture of it here on the left side of the slide. And it is a fraction of the size of an antibody. It's only about 1/15 the size of an antibody. And we have huge libraries of these proteins, and we can screen them against any target, including receptors that sit on the surface of cells. And we know that some of these receptors are really good at internalizing. They do that for a living, so to speak. That's their job in life. So we develop centurins that bind to particular receptors on the cells of interest, and because the centurin is covalently linked to the siRNA, carries the siRNA inside the cell. We've been able to show that this works in animals and mice and monkeys, and we're on our way now to the clinic later this year. I'll just show you one little piece of data. So this is a centurin specific for a receptor which is highly expressed on muscle. And we've conjugated that centurin to a silencing RNA for a housekeeping gene. It's called AHA1. And if you look at the data here, in the blue are the various muscle tissues. So we have the gastrocnemius, the quadricep, diaphragm, and heart. Kidney is yellow, liver is orange. And what we're showing here is a decrease in the gene expression of those mRNAs that the siRNA is specifically hybridizing to. So we have very nice response, dose response here, and yet we don't see any activity in the liver and very little activity in the kidney. The other remarkable thing is that this is one dose. As I mentioned, these drugs are very long-lived. So at 2 weeks, these are the results that we see, and we've gone out much longer than 2 weeks. So our lead program is targeted at a rare muscle disease called Pompe disease, and this is a disease where there's a deficiency in a metabolic enzyme called GAA, or acid alpha-glucosidase, which is the enzyme in muscle that breaks down glycogen. And without this enzyme functioning, patients with Pompe have a buildup of glycogen in their muscle, and this causes all the clinical issues that people face with progressive muscle disease. It can occur in babies, can occur in older people, and toxic glycogen buildup here is what drives the pathology. So we have identified a siRNA that specifically inhibits this enzyme called GYS1, or glycogen synthase 1, which is the enzyme responsible for synthesizing glycogen in the muscle. And our hypothesis is that by reducing synthesis, we'll be able to normalize glycogen in the muscle tissues where the breakdown is severely impaired. And we've actually been able to show this in mice because there are animal models of Pompe disease. And after a couple of years of identifying the lead molecules, we've now completed our tox studies. We've done the scale-up GMP, and we're going to be in our first human trial, amazingly enough, at an organization in Toronto in about 3 or 4 months. So that's super exciting. And we also achieved from the FDA last year the designations of orphan drug, as well as rare pediatric disease designations. So our first molecule on our way to the clinic, and we're now building up a pipeline in immunology. And we have several different programs using different receptors that are on the surface of immune cells. And the most interesting thing that we found is that when you activate immune cells, as they are activated in immune disease, if you target the right receptor, they become more receptive to these siRNAs. And this is just one experiment here shown where we have normal resting PBMCs, that's peripheral blood mononuclear cells on the left, and on the right, PBMCs from humans that have been activated. And you can see by the light blue line that when those cells are activated, we get very nice uptake and specific gene knockdown mediated by the siRNA. So we've now been able to translate this into mouse models, and don't have enough time to talk about it here, but the bottom line is that we are focused on those diseases that were not on the chart on the earlier slides. So where I want to go are diseases like Sjogren's, lupus, pemphigus, even type 1 diabetes, where the progress has really lagged versus some of the other autoimmune diseases that were highly treatable by antibodies. So that has been our journey, and we're super excited to bring this whole new class of drugs into the clinic soon. And from a company point of view, we're always thinking about what's next, what's next, what's next. Beyond muscle disease and immunology, we're very interested in using our technology to try to develop try to deliver drugs into the CNS. These could be siRNAs, they can be any kind of drug, small molecule drug, because this is obviously one of the big barriers for drugs in the industry is efficient transport and safe transport into the CNS. So we have some very early work also working on that. So just want to say a big thank you again to everybody for inviting me. It was great to be here and meet all of you. And thank you to all my colleagues at Arrow and to all my former colleagues at Centocor and Janssen who pioneered these amazing medicines that I've told you about today. So thank you.
demonstration that, that textbook slide you have, and you think of the decades of research that went into understanding that model, and then how the complementarity of the clinical medicine and the clinical experimentation sheds light on how that influences human disease and sort of moves us forward. So a great example of the basic and clinical synergy that happens as we progress along the development of those paths. So Dr. Dillon has agreed to take a few questions, if there are some in the audience. Yeah, Larry.
Great, I really enjoyed your talk. I'm from the oncology side of brain tumors, so I wonder whether you have any experience or what your thoughts are on. I think I'm loud enough, but we have some data actually coming out. I was really interested in the IL combination of 23 and 12. So we have a publication coming out hopefully this week that shows there is an immune signature of the combination of 12 and 23 that determines response to oncolytic viral therapy intratumorally, followed by immune therapy. We show that there's approximately 50% increase in overall survival for GBMs, which is an amazing result. So that actually really caught my eye, because when we did the RNA-seq analysis on the tumors to determine a biomarker of response, that combination of both being high determines the best response. So I'm curious whether you have any experience or interest to pursue looking at intratumoral delivery of the antibody.
Yeah, that's a fascinating idea. I think you're ahead of the field, because people have been so focused on the peripheral diseases, and I was not aware, actually, of the type of work that you're describing. I think that getting these antibodies into tumors, let alone in tumors in the brain, is a huge hurdle. And the antibodies themselves, as you know, don't cross efficiently. So whether hooking them to a centurion to get it across or directly injecting may turn out to be the answer. I don't know, but that's really exciting. And it's not really that surprising to me, because most of these tumors, they have a very strong inflammatory signature. And as we know, there are lots of immune cells in the brain just naturally, or cells that can perform immune function. So I'd love to see the paper. And I could connect you to people. I'm
Yes,
not
perfect.
working on it anymore, but I know people who are.
Thank you.
Yeah.
That was great, too. Thanks for Robert. Right up the spondylitis program here. So as you said, ankylosing spondylitis seems paradoxical. Why
Yeah.
should it respond to IL-17 blocker and not IL-23 blockers? So we've been interested in non-IL-23 dependent production of IL-17.
Yeah.
So there are other receptors that are druggable and targetable that may actually bypass that. But it really, in many ways, it calls into question the relationship with gut-joint trafficking, because why should 23 block the gut and not IL-17 exactly reciprocal?
Right.
So it's a great example where the clinical trials actually have led us to basic science discoveries. Usually it's the other way around.
That's right.
Fantastic.
That's right.
That textbook slide is not quite correct. Other questions? Okay, well, thank you again. Oh, sorry, one more. Yeah, go ahead, Howard.
So you're getting at the Achilles heel, or a pivotal thing in normal immune responses. So how do these patients do in terms of getting infections if they're on these inhibitors, immune suppressants,
Yeah.
probably lifelong?
Right. So that has also, I would say, with the advent of the IL-23s and IL-17s, improved tremendously versus the earlier drugs against TNF. So TNF, definitely serious infection is a risk. And with these drugs, very, very minor effect. So, that was another fortunate surprise, and it was something that we were really worried about, but it didn't materialize to any significant extent. Yeah.
Okay, well, thank Dr. Dillon once
Sure.
again. So I'd like to invite Dr. Mohit Kapoor, who will continue with the program.
Well, good morning. Thank you for the opportunity to speak at this special occasion and share Some details of the Shoulder Arthritis Institute. Can I get my slides, please? All right, as you can see from the logo, we're trying to treat osteoarthritis and more so broader arthritis with spondyloarthritis, psoriatic arthritis, lupus, and all. And what a fitting finale it would be if I actually just say that Don has been instrumental and probably the key individual who's actually been involved in promoting arthritis within Krembil at UHN. He's been instrumental in devising the strat plan and the strategies which we put in place, what is now called the Shoulder Arthritis Institute. If I have to pick words and define Don, I think, and I had to write these because I had many words to say. I think I've learned a lot myself. I started in 2014. Don started here in 2013. Don is kind and visionary and absolutely relentless, and that's the logo for Krembil. If you look at arthritis, just like neurodegenerative disorders, I think there's a lot of similarities in terms of debilitating disease. It affects millions of people worldwide, including Canada, and with a huge economic burden worldwide. We tackle with various types of arthritis, where there are more than 100 types of arthritis we tackle with, and we try to address this within the Shoulder Arthritis Institute. If you look at the history of arthritis community at UHN, it actually started almost 4.5 decades ago when the first lupus data bank was created by Dr. Murray Urowitz. Then you fast forward it from 1980s all the way to 2013 when Don was recruited at the Krembil Research Institute. If you rewind back in 2010, we actually had arthritis program, which was not an institute at that time, which was created, and also was a launch of the campaign to cure arthritis. And this timeline from 2010 to 2013 and all the way to 2020 is critical, because in 2016, Don tasked myself and Dr. Robert Inman, who's the co-director of the institute, along with Brad, with a task to create a strategic plan for arthritis. What would arthritis look like in next 10 years to create a short-term and long-term strategic research plan? He also introduced concept, along with Brad, the concept of creating an institute model, 3 institutes within the Krembil community. There was a huge process of involving various stakeholders across UHN to actually launch the Shoulder Arthritis Institute with a landmark gift from the Shoulder Foundation in 2020. Matter of fact, in October 2020, the Shoulder Arthritis Institute was launched. Our vision was very specific and well aligned with the UHN priorities to be able to create a healthier world focused on various aspects on musculoskeletal diseases from autoimmune, rheumatic, bone, joint, and spine conditions. It also led us to identify some very specific key strategic priorities which were discussed on a regular basis with Don, focused on early diagnosis and therapeutics, data analytics, translational medicine, and integrated population health. We also identified some of the enablers. And I think one of the most important aspect, and I think strength, what Don brought to the organization was creating cross-cutting platforms. And I think we have taken the note of the cross-cutting platforms, and that actually formed the basis of everything what we have done within the Shoulder Arthritis Institute. You create something within one institute, but it should be helpful for the entire organization. I'll give you some examples of those. Within the Shoulder Arthritis Institute, we integrate education, care, and research. We're over 365 individuals, over 52 clinicians and scientists, 200 staff, and 113 research trainees. We also have over 40 years of specialized research, and this is now the largest multidisciplinary arthritis institute in Canada. We're really fortunate. We have some exceptional and outstanding leaders in our 4 divisions within the institute, from hand, orthopedics, osteoporosis, and rheumatology. If you look at various cross-cutting platforms, which were done in consultation with Don, I'll give you some of the examples of things which have happened since the launch of Institute in 2020. So in October 2020, we launched the Institute, and we first launched our Biobank Center. This Biobank Center now houses over 500,000 high-quality biospecimens, which comprises of orthopedic biospecimens as well as rheumatology, various forms of arthritis from psoriatic, lupus, spondyloarthritis. In 2020, we also launched our next generation sequencing center. This is a sequencing center which provides cost-effective sequencing solutions to not only investigators within the Shoulder Arthritis Institute, but also the Krembil Brain Institute, as well as Donald K.I. Institute. In 2020, we also launched a very successful arthritis web seminar series, which brings in various international experts to talk and tackle arthritis. In 2021, we launched our Informatics Center, spearheaded by Dr. Igor Jurisica, to bring in informatics expertise within the institute, and also this also caters to the other institute within the Krembil community. We also launched the Research Training and Staff Career Development Seminar system, and this is a really good way for trainees to be trained, and a concept we learned from Don. In 2021, we launched the grants competition. So this is a PAL grant competition in line with the Krembil competitions, which Don created, in addition to the student graduate award competition, as well as the fellowship competitions, which are successfully running, and we are actually running the 3rd edition of these competitions. In addition to these, there are a few other things which have been launched since 2020. In 2023, we've launched the Metabolomics Center. We've then launched our Orthobiologics Center to test new therapies for patients with various forms of arthritis. We also ended up launching our strategic plan for education, spearheaded by Dr. Laurie Elbert and Laura Passalant. Finally, we launched our International Conference on Arthritis. This event is in partnership with the Arthritis Society of Canada, the Canadian Institute of Health Research, Institute of Musculoskeletal Health and Arthritis, as well as University of Toronto. A highly successful conference, had a first edition last year, and we are going to have the next edition next year. So if you see all these highlights within the Shoulder Arthritis Institute and you fast track, I'm just going to skip this slide and move to this particular slide, is the integration of care, education, and research. Within the Shoulder Arthritis Institute, and the concept which Don shared with us was the integration of these 3 domains under one umbrella. And this is something which is so crucial, where our clinical director, Sylvie Groh, is devised a framework where we can provide quality care to our patients. How can we create a good framework, an ideal atmosphere for our colleagues and fellows, our trainees, to actually work in an environment? So there is an initiative which is called Joy in Work, which is a UH-wide initiative, which is also led by Sylvie Groh. This particular initiative has various phases, but is focused on professional development, work-life balance, as well as clinical operation changes to be able to provide timely care as well as rapid care to our patients. All of this wouldn't have been possible because you need a lot of money to be able to have an adequate infrastructure in place. Special thanks to Annette Larson, who's the Senior Campaign Director, as well as Tennis Hansen for their immense contribution in building the Shoulder Arthritis Institute. In the past 10 years, again, on the guidance of Don, we've been able to raise over $134 million, and this is ongoing at the moment. If you actually look at all our grateful supporters, I think they've believed in the cause. What we want to do is to be able to really stop the wait time on our patients, and that's why you see no more waiting. We don't want to guess what type of arthritis you have. We want no more guessing game, and we want to eliminate arthritis, and that's why you see no more arthritis. And with all of this, if you're really interested in to know a little bit more about the Arthritis Institute, please visit our website or subscribe to our quarterly newsletters or follow us on social media accounts, whichever would know. And finally, I think the vision which Don had, that 3 institutes can actually not only exist under one umbrella, which is the Kreml organization, but actually thrive and be world leaders in the area of research. I think it's a vision which very few people have, and we are grateful that we've been able to realize this vision and a dream to create the Schroeder Arthritis Institute within the Kreml community. And also my special thanks, along with Don, to Brad and other leaders within UHN who really realized that arthritis, or tackling arthritis, is important. And thank you so much for listening to me. All right, well, switching gears back to neurodegenerative diseases, it is my pleasure to welcome Dr. Barbara Tate, our next guest speaker. Dr. Tate has been the Chief Scientific Officer of the Friedreich Ataxia Research Alliance since spring of 2020. Previously, she was the Chief of Strategy and a venture partner at the Dementia Discovery Fund at SV Life Sciences. She has worked in both large and small pharmaceutical companies where she has held executive positions. She was the founder and the CEO of Tiaki Therapeutics. She also was a founder and vice president and head of biology at Rodin Therapeutics. Prior to Rodin, Barbara was a vice president for research at Satori Pharmaceuticals, and at Pfizer, Barbara managed the neurodegenerative area group in Groton. Prior to working in industry, Barbara was an academic researcher at Brown Medical School and Harvard Medical School. Barbara has a BA in biology from Beaver College, PhD in physiology from the University of Delaware, and completed her postdoctoral fellow at Harvard Medical School. Welcome, Barbara.
Thank you. I'm very happy to be here. I'm a little bit surprised. I'll tell you how I got to meet Don. Treventis, which is one of the companies that he founded, had a Wellcome Trust grant. And the Wellcome Trust really didn't want to fly back and forth from London frequently, so they asked if I, coming from Boston, could be the representative for the Wellcome Trust on the Scientific Advisory Council for Treventis. And I have to say, I was tough. I was really tough on the company. You know, they're a small molecule. You know, the chemistry was very strong, but I, as a biologist, often said, you know, good chemistry can rescue bad biology, but if we don't understand the biology, we're really not going in the right direction. So pushed hard. So I'm really pleased that Don thought that was a good thing, and that I'm here today to talk with you. I will tell you that what I'm going to talk about, and it's a very weird experience for me. I think I skipped a slide there. Oh, these are my older slides. These aren't the ones that we were going to replace with. All right, so I'm going to have to just wing it on a few of these slides. Why am I here? You know, I'm not going to present some of my own data slides, which is a really weird experience for a scientist. But I did have decades of working in the lab, jealous in some ways that Don's going to be able to go back and do that. And also, as you heard in my bio, decades of experience in leading, and finally, a career in venture investing. So that gives me a perspective, I think, across the landscape of drug development and discovery, and really gave me some insights that I hope I can share with you today. In the last 3 years, I've been supporting a rare disease, a monogenic rare disease, Friedreich's ataxia, again to do discovery and development of therapeutics. And I hope that experience in the rare disease actually can reflect on these more common diseases and teach us fundamental things about the common diseases. These are my own personal opinions. and shouldn't be attributed to my employers. So why do we need new directions for treatments for Alzheimer's disease? As you know, we've gotten a few approvals. So very rare in this field. We hadn't had approvals for a number of years, and then quickly 2 drugs were given accelerated approval by the FDA. But I want to make the case that those drugs really address one pathological feature of the disease. And this is a very complex and varied disease, varied in clinical presentation, varied in the underlying neuropathological features of the disease. And so while we have these recent therapeutic approaches, they were based on a prevailing hypothesis a number of years ago that was very precise. So we're looking at a disease that's very varied, both in clinical presentation and neuropathological features, and a very precise hypothesis that was driving our therapeutic approaches. Our recent insights is that this is probably not the right way to think about Alzheimer's disease. In fact, it's a disease of multiple processes that underlie the common forms of disease. And we know that, in fact, if we look at the therapeutic benefit of the recently approved expensive and difficult to take therapeutics, it's limited. And it's limited both in impact and to few patients. So we really don't have those therapeutics that are going to address the variety of Alzheimer's disease and benefit as many people as we possibly can. Yet we're spending a lot of money on those therapeutics. So how did this come about? I'm going to give you a little bit of a history lesson. I'm sure you're familiar with most of this. Alzheimer's disease is named for a German neuropathologist from the early 20th century. He had a very famous patient, Augusta Dieter, shown here, who represented to him the disease process. But in fact, if you look at some of the characteristics of this woman, it was very different than what we consider our more common forms of Alzheimer's disease. For one thing, she was 51 years old when she passed. And you'll know, as you're familiar with, Alzheimer's is a disease of those in their 70s, 80s, and even 90s. So that's a fundamental difference. She also, while displayed the memory and language deficits that we think of as common in the disease, she had behavioral aspects and other psychiatric features that are not common in Alzheimer's disease. So while this is thought of as a representative patient, in fact, it's not representative. She is not representative at all of most Alzheimer patients. And that's really, I think, reflected in that Alzheimer gave her a diagnosis of pre-senile dementia. So not what we think of as our more common forms. What he did describe were the neuropathological features of the disease, and they're both extracellular and intracellular features that are commonly seen and diagnostic in postmortem analysis of Alzheimer's disease. So we have extracellular plaques, which we now understand, so this is one of Alzheimer's original drawings. We now understand those plaques are really an accumulation of a protein and an amyloid conformation made up mostly of the A-beta protein, which is a small protein that's clipped from a larger molecule called the amyloid precursor protein. Now that protein is expressed in the brain ubiquitously and through development seems to have some function. But we were so focused on amyloid that we've called it the amyloid precursor protein and ignore that it might have a physiological function beyond that. The intracellular features of the disease are tangles, or a hyperphosphorylated form of a cytoskeletal protein, tau, that forms within the neurons. And eventually the neurons die and we have ghost tangles that are left behind. A contemporary of Alzheimer, who does not get quite as much press, Oscar Fischer, described many more patients. He looked at more patients than Alzheimer's did, and he described across that landscape that in fact there's a variety of pathological features that are seen in the disease. And in fact, there can be patients that present with dementia that only have plaques, or patients that present with dementia that only have tau. And I will point out that Don Weaver received the Oscar Fisher Silver Award in 2022, keeping us very honest about what we understand about the pathological processes in these neurodegenerative diseases. But how did we get here? How did we get to this amyloid cascade hypothesis? So this was pushed very heavily within the field, and this is the precise hypothesis that I was talking about earlier. Very precise, that there's a direct line from amyloid to dementia. And it goes through a number of steps, but each of those steps is really driven by amyloid itself. So this was based on some genetics, saying that there were mutations in both that amyloid precursor protein and in enzymes that clip the amyloid precursor protein, presenilin 1 and 2, to result in A beta production. So that is genetics that would point us in the direction here. And then we see the ongoing accumulation of amyloid in the brain. And then there were some, I would say, hand waving that came beyond that to show that there were injuries that resulted from amyloid itself. And Selko and Hardy in 2002 expressed this in definitive terms. According to the amyloid hypothesis, accumulation of A beta in the brain is the primary influence driving AD pathogenesis. The rest of the disease process, including formation of neurofibrillary tangles containing tau protein, is proposed to result from an imbalance between A beta production and A beta clearance. And this led to diagrams like this, where we have those pathological features, amyloid plaques and neurofibrillary tangles, and we stage the disease by the quantity of changes. This is all post-mortem analysis. We weren't at this time able to actually look in the brain of patients, but in post-mortem analysis, we staged the pathogenesis as a stage of the disease. So more plaque, more tangles, were more severe forms of the disease. Clearance, remember we said, was a therapeutic approach that Selko and Hardy suggested. Production as well. Production turned out to be a lot harder. Those enzymes are difficult to modulate safely. But clearance we did see in antibodies, and I was so grateful that we now know what antibodies are, thanks to Sue, that antibodies could in fact clear amyloid from the brain in preclinical models. Mice that were transgenic, overexpressing the human APP protein and producing lots of amyloid in the brain. Lots of amyloid, something like 64-fold what we see in normal, in humans. I want to point out there was never a good correlation between the amount of amyloid in the brain and the clinical manifestations of the disease. That never existed. But we still went on and produced antibodies that were specific and went into very large, very expensive clinical trials. So I'll tell you about an antibody that I worked on, bapineuzumab, acquired many times by Pfizer, where I was in the neurodegenerative division. This antibody targets just a portion of the A beta peptide, the N-terminus. It has to be delivered, as they all do, intravenously. This was a very long trial, 18 months, and a large number of people. So 2,400 people participated in this trial. In fact, they also subgrouped. There are other genetic risk factors. Those very, very rare forms of Alzheimer's that are mutations within APP or PS1 or 2, they do not represent many people at all. But more commonly, there's a risk factor in the ApoE4, ApoE protein in the E4 allele that increases your risk of developing Alzheimer's. It's not definitive, but it does increase your risk. So the thought was, well, let's separate those two groups of people in the clinical trial. We'll have people that are carriers of ApoE4 and people that are not carriers. Bottom line, it didn't work. There was no improvement in cognition. There was no improvement in function. We did very large, the last phase 3 trial was $650 million US, and yet nothing. We saw no improvement. People have done retrospectives and tried to parse through, so dividing up the, these are 2 clinical measures, ADAS-Cog and the DAT, dividing up by carrier or non-carrier, do we see anything? Is there any signal here? And in fact, at a higher dose of bapineuzumab, people did worse. So this stopped. And although it did not, in fact, impact the theory. So there was a lot of hand-wringing. Pfizer certainly spent a lot of money in doing these trials and a lot of questioning as to what could go wrong. And one of the things that was very helpful was developing, instead of the clinical manifestations of the disease, actually trying to look at the biological basis of the disease, trying to then image people and look to see if we could see changes that were occurring in the brain that might help us understand the disease better. And one of those innovations was a PET ligand for amyloid. So we could look at people with the PET ligand. That was done in the bapineuzumab trial, but there's a cutoff where we see a signal that's probably just noise. It's probably not sufficient amyloid to be driving disease process. It turns out retrospectively, when we look, about a third of the people enrolled in the clinical trial for bapineuzumab did not have amyloid in their brain by the imaging endpoint. They did meet clinical criteria. These are some of the best physician scientists in the world that were diagnosing these patients for enrollment in a trial, and yet there was no amyloid that was driving that clinical manifestation. So, and it didn't matter whether you were ApoE4 or APO or not. There was such a spread in the amyloid deposition that in retrospect, having a third of patients in the trial that didn't have the target and having this kind of spread in the target is probably, even at powering with 2,400 people in the trial, was probably doomed. While we were doing imaging, we also were, I think we're good. I'll make it up. While we were imaging the brains, we discovered there were changes in the brain that got the acronym of ARIA, amyloid-related imaging abnormalities, that were discovered by doing repeated MRIs on the brain. And there are 2 forms of this hyperintensity that's seen on MRI. One looks like vascular edema, so something leaking from the blood vessels. The other is actually a microhemorrhage. So there's ARIAs A and ARIAs E. And unfortunately, the ARIAs were associated with side effects. So they were not benign in every case. There was also a piece of data that always intrigued me. So the antibody trials that we're talking about, bapineuzumab, is a passive immunization. So antibodies were made, humanized, and injected. But prior to that, Elan Pharmaceuticals had done an active immunization trial. So they had injected A-beta into people and raised antibodies to that A-beta protein. We know that they did raise antibodies. But again, no cognitive improvement, no functional improvement. Everybody in that trial, the AN 1792 trial, died of Alzheimer's disease. A lot of the brains did come to autopsy years later, and we see while there were no changes in cognitive function, there were brains that came to autopsy that had no amyloid. So the passive immunization might have worked to clear amyloid, or it could have been these were also misdiagnosed patients. As we said, we can misdiagnose people as having Alzheimer's disease when in fact they do not have the hallmark pathological features of Alzheimer's disease. Again, there's a lot of hand-wringing about why did bapineuzumab fail. Amyloid was seen as, I've heard people say, the golden target. So how could we have had the golden target? We were actively clearing amyloid from the brain, and yet we did not have a clinical impact. Many people suggested treatment was initiated too late. If we look at the amyloid cascade, neurodegeneration is at the bottom of that cascade. And we do not believe, in fact, the amyloid proponents did not believe that we were going to reverse frank neurodegeneration. If we'd lost neurons, we weren't going to replace those. So maybe it was just too late. We could clear amyloid, but we weren't going to reverse the downstream consequences. There was also the suspicion that bapineuzumab was binding. We knew it was binding both the monomer, A beta, and the amyloid. And maybe the monomer actually does have a physiological function. Maybe that's a bad thing, to bind the monomer. Also, those side effects that we talked about were not benign. Having ARIAs, in some cases, caused irreversible damage to the brain. And bapineuzumab drove those ARIAs pretty strong, so it was actually the only dose that could go forward was a low dose. Maybe it was just too low. So lots of companies became interested then in generating our next generation of amyloid antibodies. And I've circled two here that we'll talk about a little bit more. These were focused very much on what form were we going to go after. Did we want to spare the monomer and just go after the aggregated form of A beta, the amyloid? And these were very potent antibodies to go after amyloid and not potent against the monomer. So amyloid clearance did occur. We can see that in PET imaging. Both of these antibodies actually were given accelerated approval, which means that there was a biomarker change, but it's not based on the clinical impact. And now they need to do post-approval studies to demonstrate clinical impact of these drugs. They're very expensive and they're hard to take. These are monthly or every 6-week infusions, going to an infusion center and being infused. Both of these, while approved, the treatment effect is modest. And the clinical meaningfulness in the field is being hotly debated. These people were progressing, remained demented. There was no improvement, but there was a slowing versus placebo or natural history studies. ApoE4 homozygous, so having 2 of the ApoE4 alleles, puts you at high risk of Alzheimer's disease, the highest risk factor that we know of. And yet, these are the patients that are also developing ARIAs. So they might not, in some cases, physicians will not prescribe them the antibodies. So they're left out of being treated. There are other safety concerns as well. If you're on blood thinners, it appears that you may be at risk for the hemorrhages that occur. Many of our elderly patients are on blood thinners. So this is not going to help everybody, and it's not the therapeutic benefit that we have all hoped for. There's also, I think, something else in the disease that is really important for us to appreciate. And I was struck by forms of arthritis, being able to say forms of arthritis. One of my missions is being able to say forms of Alzheimer's disease. We've been lumpers, and I had very robust disagreements with some of the commercial colleagues at Pfizer because, they were looking at a $16 billion market if we could treat Alzheimer's disease. And I was trying to say it's 16 $1 billion markets. I think that if we look at the heterogeneity of the disease, and this is a recent slide from the ADNI group, which is a large consortium started out as neuroimaging, but they've really gotten into biomarkers and other stratifications of the disease. Everybody at the beginning of these graphs has what we call mild cognitive impairment. So there's been this push, let's go earlier. If we have frank neurodegeneration and we're not going to reverse that, let's go earlier in the disease. Well, earlier we get to subjective measures of cognitive impairment. We're not very good at measuring brain function, especially cognition, with robust measures. So it becomes subjective. So mild cognitive impairment is everybody who's been sleep deprived, you travel a lot, or you have young babies in the house, you've experienced mild cognitive impairment. Why do I come into this room? I forget why I'm here. I forget what day it is. That is what people are experiencing. But if you look at what happens to those patients over time, this is a 5-year period, a number of them don't progress. They presented clinically the same way, but they don't progress. And then there are some that have a very rapid deterioration in cognition. But we don't have any way of separating those right now. We're not sophisticated enough to really understand what's going on. But there is a clue. When we look at these common forms of Alzheimer's disease, we see plaques and tangles, but we see a lot of other things too. The comorbidities or co-representations of other kinds of pathology is much more common than a pure disease like Augusta Dieter, who was, remember, 51 years old. The older we get, the more these other pathological features appear. And some of these alone will cause dementia. So Lewy body dementia is a form of dementia. In fact, dementia with hallucinations, maybe Augusta Dieter actually had some Lewy body disease. TDP-43 also presents as a pure frontal temporal dementia. But TDP-43 accumulations in Alzheimer's brains are common. And vascular dementia, deposition of amyloid in the vasculature, very common as well, especially in older patients, but alone can account for a demented phenotype. So the common forms of Alzheimer's disease present with these other pathological features, and we know that these are, this is where I'm going to be sad that I'm missing my other slides, because my Treventis slide, I think, is not here. The presence of Alzheimer's disease is not deterministic. So we know that you can have amyloid in the brain, and you do not have Alzheimer's disease. We also know that you cannot have amyloid in the brain and present with the neurological features of Alzheimer's disease. These other pathologies are very common. Remember that most of our patients, unlike Augusta Dieter, they're 30 years older than Augusta Dieter. By the time we get into our 80s, there are other pathological features, and aging overlaid on these pathologies really make a very complex picture to address. And it is at least the association of having those additional neuropathological features does increase the severity of cognitive impairment. So they are playing into the final clinical picture that we see. So what has happened in the field? Well, thankfully, the field has really adjusted to this understanding that what we call Alzheimer's disease represents a vast diversity of both clinical representation but underlying neuropathology as well. And so a group like Cures Alzheimer's, which is a large philanthropic organization, presents in their target space that they're very interested in funding many more features of the disease than just amyloid alone. They still have amyloid on there, but they're also representing tangles and inflammation. And one of the features I want to point out, knowing that you know that I work on a monogenic disease, is look at all the genetic risk factors. There's over a dozen genes that have been associated with risk for Alzheimer's disease. Working on a monogenic disease, that really bothers me. How can all of these genetic factors really be aligning on one particular focused hypothesis? That just doesn't seem biologically relevant. So I think we need to think more broadly about who are we treating, and what is it that's underlying, that's the driver, the primary driver for their version of what we call Alzheimer's disease. The 100+ study has been really helpful in understanding these are centenarians that go on to be cognitively, it's still functional. Many of them do have deficits, but when they come to autopsy, we see this range. And this is the study that says amyloid is not deterministic. By the time you're in your 90s, you're going to have amyloid in your brain. That does not mean you're going to have Alzheimer's disease. There are other factors. One of those other factors is tauopathy. So we looked earlier on this slide, and we saw that we started to stage the disease by the presence of amyloid and by the presence of tau. The sort of dirty little secret is that tau always tracked with cognition in a way that was predictable. Amyloid does not. And maybe my tremendous slide is still in here. Nope. Oh, there it is. Okay, that's all you get. So what I was excited about when I joined the Treventis team was that they were not only focused on tau, which I've told you is much more, is actually correlated with disease progression, but they were focused on a small molecule approach. You know, having to go in and get $150,000 US dollar infusion periodically through the year, this is not equity, right? This is going to eliminate a number of patients from being able to be treated. Small molecule approaches do reach the masses. This is a way that we can actually address the disease. And it's possible that the tauopathy that underlies, or that it's present in Alzheimer's disease is more important in driving the physiological features or the clinical features of the disease. So I was excited about a potentially disease-modifying approach for Alzheimer's disease, but certainly for those tauopathies, pure tau deposition driving neurodegeneration. And recently, Treventis signed a partnership with Takeda. So fingers crossed that really gets this to more people quickly. So our new hypotheses really are based on there's multiple pathology in the brain. These are elderly brains. We need cocktail approaches, and symptomatics are important as well. You heard that I am still a venture partner, very part-time, at the Dementia Discovery Fund. So this fund was founded specifically to be outside of the amyloid space. These were pharmaceutical companies that came together and said, I've got lots of approaches for amyloid, but I don't have anything else. Let's use venture as a way of developing those other approaches. And if you look at the portfolio of the Dementia Discovery Fund, you don't see any amyloid approaches in there. These are approaches that are across other domains. Synaptic inflammation, both microglia and astrocytes, protein degraders, which is a way of getting at tau that may or may not succeed, but you know, much more diverse approaches that I think are finally embracing the fact that this is a multifactorial and complex disease. The other thing that's very important, I think, in succeeding in trials is that we really do have to understand that the clinical features are not predicting the patient population. They're not giving us insight into what we actually want to treat. We want to treat the molecular basis, the underlying neuropathology. But what has occurred in the field, thanks to ADNI and other large consortiums like that, is that we've got better biomarkers that we can use now. We can measure A-beta in plasma and CSF. We can measure tau. We can do imaging as well. We can measure inflammation, not as well as we hope, but we're getting closer to that. And we can think about neurodegeneration. We can look at structural changes in the brain that actually reflect neurodegeneration. This recent paper suggests if we want to succeed in these clinical trials, we really do need to parse patients by that underlying pathology. All comers Alzheimer's trials will fail. I will say that unequivocally. All-comer Alzheimer trials will fail. We really need to get to a more clinically relevant, we'll say, pathological feature of the disease that we want to treat. One of the challenges, of course, is which pathological feature is driving the most pathology, and that we still are parsing apart. But all comers will not work. So what is the future state? I hope the future state is that we do have clinical diagnoses. This is still a big gap for us, clinical diagnoses that reflect the underlying pathology. We really do need to take a step back and get outcome measures and diagnostics, biomarker, but also clinical diagnostics that are much more specific about what is happening to that patient. ADAS-Cog doesn't get us there. We have to really embrace the fact that there are age-related changes that are occurring in the brain. There is a difference between a 50-year-old brain and a 90-year-old brain. And if we don't embrace that, we're probably ignoring some of the factors that are at least influencing disease progression in that patient, and maybe be treatable, but certainly are going to interfere with our ability to parse out, as we maybe could have in Augusta Dieter, where she was really 51 and had pathology, from what is happening and our therapeutic benefit. And honestly, I think the magic bullet isn't there. I think that the number of pathologies that we see in an aged brain is really necessitates that we need to come after this as we have done in other complex diseases. There are a number of drivers here. I'm going to use the oncology analogy. There are drivers here that we need to address holistically, and they may change over time, even within a single patient. So a complex cocktail approach. So thank you, and thank you, Don, for inviting me today. We have time.
Fantastic lecture, Dr.
Yes.
We'll take a few questions. Go ahead, Mary.
Thank you very much. That was wonderful, and definitely your opinions align with mine. I'm wondering if we can go back to the idea about clinical informing the basic science.
Yeah.
And there's a lot of interest in about modifiable risk factors in Alzheimer's disease, maybe even in preventing, but at least slowing progression, et cetera. And I wonder if there's anything you can take away from those that might feed back into the kind of pathological space, or are we just kind of, not just, but are we treating, the symptoms and the disease marching on?
Right, yeah, no, it's a very good point. And there are data, and I think this came out of ADNI, but I'd have to check, that we have seen a decrease in diagnosis for Alzheimer's disease. And it was probably because at least some generations, my parents were eating red meat every day, and our generation is not doing that. And vascular disease is an addressable risk factor for Alzheimer's disease. And I think as we have addressed that, we've actually seen an improvement. So I agree. I think there are modifiable components of the disease, which is why I'm running a marathon on Sunday. And maybe it helps with arthritis too, I don't know. But I do think we know, in some ways we know that, that healthy lifestyle does change the course, or at least the rate of progression.
But there's no data yet to say that it changes tau, or as far as you're aware.
Right. Unfortunately, these are, again, we're looking at population studies and we're looking at Alzheimer's disease. In the tauopathies, I don't think there is data that there are modifiable risk factors. Probably genetic, though, probably, multifactorial and some genetic, SNPs that are protective.
Antonio and then Rob.
Yeah, Barbara, great presentation, thank you. I'm a neurologist, movement disorder neurologist, and whatever you said today on Alzheimer can be replaced with Parkinson and Parkinsonism. Exactly the same controversies and problems. And again, you can replace amyloid with alpha-synuclein. It's exactly the same issue, right? So you can have Parkinson with no alpha-synuclein. And so the problem is, again, we had the same challenges with no success in clinical trials for the same reason. Of course, we are stratifying our patients based on clinical features. I'm a neurologist. Of course, we stratify these patients based on their presentation. But again, the importance, as you mentioned, are the biomarkers. And probably, even to early disease, it doesn't really matter at this point because you cannot really identify patients when they don't have a disease, right? So you need a good biomarkers, and even one biomarker is not good enough for
That's right.
progression and diagnosis, and also for, so definitely I think all the future clinical trials will not succeed unless we have a good biomarkers, and that is the key, right? Because clinical certification is not going to work now in the future, even if you have a successful drugs. So you cannot have these patients all together, you just give the drug and see what happens. You need the biomarkers, and I think this applies to ALS, Parkinson, Alzheimer's, the same. So thank
I
you.
agree with you. The tragedy is not a failed clinical trial. To me, a trial where the outcome was clear. It didn't work or it did work. That's a good trial. That's what we need to do. The trials that we should be embarrassed about are ones like aducanumab, the original trials where we don't know whether it worked or not. That's unacceptable. Right? And I think that's because we haven't really, as much as we want to say the clinicians and the basic scientists are talking to one another, it's now that we're catching up and saying there's a biological difference between these patients that I'm underappreciating, and that I was really going to change the outcome of my trial if I don't pay attention to that. At least in Parkinson's, I think there's more of an embracing of genetic forms of the disease, and that's hugely helpful. In Alzheimer's, we just haven't been able to sort that yet to see if there are, there's tauopathies, but are there other genetic forms of the disease that we're lumping together and saying they're actually the same disease when they're not?
Dear Dr. Tate, it was a wonderful presentation. My name is Gabor Kovács. I work as a neuropathologist, a neurologist here. And as a neuropathologist, I had the privilege to look through in the last 2 decades more than 5,000 brains with the standardized methods, and I'm very happy to hear your approach and confirm that in a 100 years period I haven't seen any brain, or very rarely, pure Alzheimer's disease brains. But I also see variability in the distribution of the tau and the peptide composition of the beta amyloid between patients. So my question is to elaborate on your approach, on cocktail approach, that even for tau, for example, if you want to focus on tau, we see differences in the tau isoforms and tau strategies, and also we see that the mRNA production in the cells are preserved or even increased in the pathological state. So if you approach a cocktail approach, can you envision that you not only approach from different proteins and immunological approaches like pathogenesis related, but also the same protein you target from several ways. Like for tau, you decrease the mRNA expression entering the cells, not to feed the seeding of the bad tau, but also to target the bad tau. So can you envision in a cocktail approach targeting the same protein from different pathways?
Yeah.
That's my question.
Yeah. No, I think that's a great idea. I think converting that theory into practice is the challenge, right? That we put ourselves in the amyloid box because of the resources that were necessary to be able to actually execute those trials. They were huge, billions of dollars, honestly, went into that. And I think if we can get better at executing the trials so that we can iterate in people, mice lie and monkeys don't tell you the truth. You get so far in those preclinical models, but you really do need the human experience to inform you. But we can't iterate in those trials now because they're just too expensive to do. So I think biomarkers are the key. If we can really get to decision-making biomarkers and appreciate, amyloid isn't changing, I need to back off, or tau, it's going in the opposite direction, and I don't understand that. That's what we have to be willing to do. And I think you see that more in the monogenic diseases, where you do know that you have to change in frataxin increases or you're not going to get the clinical benefit that you want, you can make a decision. But we haven't embraced that in Alzheimer's disease. Even when we knew we were going in the wrong direction, gamma secretase inhibitors, we still did the trial.
Okay, I think we're going to take one last question and then we'll have a coffee break so we can have.
Barb, that was great. I was struck by the heterogeneity of the clinical trajectories. Taking a page from the arthritis book, in diseases where the natural history is that variable, we've encountered significant issues with FDA on what constitutes a placebo. What is the ethically acceptable trial design for a placebo arm? Because we're eventually going to compare intervention versus placebo. So what have you encountered? Is that a significant challenge in trial design?
No, it's huge. It's huge. This is why, after spending decades working in Alzheimer's disease, I had the opportunity to work in this monogenic disease, and I was like, yes, please, because there's a 20-year natural history study, and even in this monogenic disease, people present differently clinically. So it was there was a recent approval. The first drug for Friedreich's ataxia was just approved, and part of it was on looking at natural history, placebo differences, and understanding that we could match one-to-one the right patients that were in the trial with the right natural history patient, because it isn't the same. So I worry that if we don't get better at large diseases like Alzheimer's, at understanding, and the genetics is driving that in Parkinson's disease, where we don't do GBH and alpha-synuclein in the same trial now, if we don't get better at doing that, we just can't afford to run the trials that we need to run, because you're going to have placebo groups that are hundreds and hundreds of people.
Okay, well, thank you very much. We're going to take a 10-minute coffee break, bio break, and I'd like to just invite all the speakers, so if you're speaking at some point today, please come up for a photo. I'll catch up. I saw your email. Okay, it looks like everyone is having a great time. I'm sorry to disrupt all the conversation, but I'll ask you to find your seating. There we go. So I'm sorry to interrupt all the conversation going on. Looks like everyone's having a great time. This doesn't happen on Zoom. But please just ask you to find your seats again for the remainder of this morning's program. Okay, it gives me great pleasure to introduce UHN's Chief Executive Officer, Dr. Kevin Smith.
Thanks, Brad. Thank you very much. Thank you. It's really looks like a great morning. I got to hear a little bit of the last speaker and really enjoyed hearing how common the discussions are across many neurodegenerative diseases. And by unlocking one, we're going to unlock a lot more. So thank you very much. I'm really sorry I missed the birth and evolution of Krembil. Where did he go? I wanted to apply that to Krembil himself as opposed to just the Institute. But I'm very, very pleased to be with you here today to celebrate the leadership and indelible contributions of Don Weaver as a key member of Team UHN. When I think a bit about what does it mean to be an ideal team member at UHN, Don really jumps to mind, not only because he's a renowned neurologist and scientist, but because he's an exceptional leader and teacher. And he's not enjoying this one bit because he will not look up from the table. As you all know, Don is a remarkable person. He's remarkable also in that he's decided at this point in his career to focus on what might make a difference to patients and scaled back on everything else because at the root of making difference to patients is basic science. And never more than this week, as we saw yesterday in some of the challenges of science funding, has that been clear. So Don, thank you for doing that. Don, as you know, an internationally renowned expert in drug discovery and research in Alzheimer's and dementia diseases. Don has helped both elevate UHN and Canada's profile as a destination for discovery and a destination for commercialization, which of course is a very important ingredient in how we take this literally around the world and find models of funding it. A rare combination of a neurologist and a PhD medical chemist, Don's research, as you know, spans basic discovery and drug design to clinical trials in epilepsy, dementia, and Alzheimer's disease. That truly is remarkable. Don, you are the definition of bench to bedside or bench to patient side. And yesterday, Don shared something that I hope you don't mind if I repeat here today. Don shared with the group, a few of us last evening got together to thank the Krembil family as well as to recognize Don. But he said, I've told 10,000 people they have Alzheimer's disease. And unfortunately, I've had to tell those same 10,000 people that I don't have something that can really help cure that disease for them. And that for me really says it all. That why are we doing this? Back to those people every day who are hearing a devastating diagnosis like that. For many of us, the most frightening diagnosis. Not only because we lose our physical health, but because we lose our identity. And for so many of us, it's our greatest fear. Don, as you know, has published in many popular methods for computer-aided drug design, the co-designer and co-developer of 2 drugs that have reached international phase 3 trials. He's co-founded 8 biotech companies to translate his research to patients. That's pretty remarkable. He's also a pioneer of biotech in the concept of micropharma, and academia or originated biotech startups are small, efficient, and patient-focused when they're done at their very best. Don takes all of his responsibilities very seriously, but none more so than training the next generation of providers and those who undertake drug discovery research. Listen to a couple of statistics. 49 postdoctoral research fellows, 25 PhD students, 21 master's students, Don, you've seeded a small university, so thank you for that. And of course, over his career, Dr. Weaver has had many prestigious awards, including most recently the Oscar Fisher Prize for Innovation in Alzheimer's Research. He's accomplished all of this while practicing, and more, while practicing as a neurologist and leading the evolution of the Krembil Research Institute. And truly, that's nothing short of amazing, and why we're here to celebrate Don and celebrate science today. But luckily, Don is not done yet, so luckily your shoulders are broad, and we look forward to seeing what research and amazing accomplishments come next in your tireless efforts in research and in changing the life of people suffering from Alzheimer's disease. With that, I'll now turn the podium over to Dr. Andrews, the Krembil Trainee Affairs Committee Chair, who will speak about Don's impact on trainee culture. Thank you very much. Don, congratulations.
Okay, thank you. I get the distinct opportunity and pleasure to be able to talk a little bit about Don's exceptional work as a mentor to many of us in this institution. So we've already heard about Don by the numbers, which is the kind of static picture of him. He's a remarkable experience with training the next generation and maybe the next generation after that of scientists. But more importantly, I was able to dig into his 194-page CV in order to find some nuggets about his training philosophy. So he starts it with the mind is a fire to be kindled, not a vessel to be filled, which is a lovely quote, I think, from Plutarch. And I think some of these examples down below also show you some of that philosophy operationalized, that he really thinks that mentorship or learning experience is dynamic and mutually responsive, that the lessons that he delivers are focused on what he calls the 3 E's. And of course, education we all know about, but only Don can deliver that distinct character of engagement and particularly entertainment, I'm sure, in his classes that we all know about. And again, the idea of wielding multiple ideas and concepts, sometimes from left field, but many times enriched by both his clinical and basic science practices to be able to pull these things together, and his poetry, as we've heard, and probably will hear more about. I also asked his trainees to tell me something about Don. Give me some words, and we can make a word cloud out of it. And I think you can see some of these characteristics: inspiring, determined, passionate, relentless. I don't think made it in there, unfortunately. But I think importantly from the trainees' perspective, supportive takes front and center. And I think that's what most of his trainees have felt about his experiences with him. So here's Don. This is the way we always think of scientists, you know, in the lab, instructing, here's what we really need to do, et cetera. But that's only a small slice of what Don is. Don is also relentless in engaging his lab members in the annual hockey tournament, where I understand he gives no quarter to anyone and is quite pleased to be able to conquer them all in that experience too. I think importantly for me too, Don is really good at being able to show that science advocacy is something critically important. We heard earlier about some of the outreach kinds of opportunities in terms of magazines, et cetera, but really getting to the general public and say, why does science really matter, and why should we enable individuals to support this? So he's a role model in that respect, and maybe many of you don't actually appreciate that Don is actually a sartorial role model and tells people that, you know, this is the way to succeed if you want to just dress in the khakis, the tie, and not the tie, and the striped shirt. So we all appreciate that about Don. And so that's his lab, and one thing that all of his lab members also say is that Don's always trying to elevate everyone. So as you can see from this picture, just the idea of not only jumping for joy, but also making sure that those trainees are going to be able to fly on to more success in the future. But Don is not only responsible for his own lab. You saw it was a pretty big one, so that would be enough for most of us to be able to handle. But in fact, as the director of the Krembil Research Institute, he is actually overarchingly responsible for 3 different pillars: neuroscience, arthritis, and vision, as you heard today. So how does he foster some of those things? Well, Jadip actually stole some of my thunder, but that's okay, by telling you about some of the initiatives that he launched. But one of the major kinds of initiatives that happen every year, and that has really been boosted, I think, a lot by Don, Don, is our Krembil Research Day. So I just wanted to show you a couple of pictures from that. And really the important part is not only has it grown during Don's tenure here, from 184 registrants in 2014 to 277 in 2019. Why did I stop there? Well, the world stopped there because we usually have it in June. But nonetheless, both he and the wonderful administrative team that he has with him managed to keep us going, even in the world that we now all appreciate and know throughout the pandemic, to be able to kind of keep the science alive, because that ability to kind of come together as a scientific community is what really enriches the students' experience here outside of your own lab. So, what would Don have to say about Research Day in his own words? Cue those guys.
Krembil Research Day is an opportunity for the trainees that we have at the Krembil Research Institute to present their work and discuss the exciting science and research activities that they are pursuing. This is an opportunity for people to share their ideas, share their research, interact and mingle. Also, because it is trainee focused, it gives them an opportunity to present in a setting where they may be challenged on their ideas and they get to defend them. And this is a good learning opportunity.
Great learning opportunity, and thanks to our impeccable Krembil videographer team for putting that together. So what does Don also like? Don loves to be able to give out prizes to people. So these research days and the summer students' research days allow him to actually let students not only defend their ideas, but also feel that wonderful sense of accomplishment of being able to kind of come in with the first prize. And we heard a little bit, or at least we saw a snippet about one of the ideas, I think from his experience in commercialization, that he really brought to the Krembil trainees, which is, how do you do an elevator pitch? How do you convince somebody who knows nothing about your area of research that it's interesting, intriguing, and something that you'd really like to be able to invest in, hopefully? But it's not all hard work. Don actually loves to be the center of attention at many different kinds of events and usually makes a competition out of them as well. So for here, we have the can day where we were getting a food drive, but we also had to be able to build some kind of clever little monstrosity. In the pumpkin carving contest that he initiated, as well as in the, not so annual anymore, but the ugly sweater day. It's hard to demonstrate an ugly sweater on video, but at any rate, the ugly sweater day has always been a nice Christmas institution. Not all hard work at all. Okay, so now you will have to excuse me because I have a little hoarse voice, but I wanted to give a poetry tribute to Don as well. I'm not a poet, accomplished poet like Don is, but occasionally I've been known to do things like take song lyrics and mix them up a bit to see if we can make them be appropriate representative. So here, this is probably something that Don would have remembered from his past. Apologies to Crosby, Stills, Nash & Young for adapting their Teach Your Children Well. Don, all along this road, has had a code that he has lived by, and this he believes is key, inspiring minds to question how and why. Teach your trainees well, unleash their dreams, ambitions and passions. Give them tools to excel, igniting fires, coupled with compassion. There's only the chorus left, it's okay. He's their mentor and their guide, and he looks on them with pride as they become themselves and fly. Ooh, that one. Now that's some legacy. Thank you. So thank you, Don. Thank you from all of us. Thank you. And trust me, I would not do that for many people. So So much appreciation from all of us. And now I get the distinct pleasure to actually introduce one of Dawn's trainees, Sydney Wyszynska, to give sorry about that, Cindy, to give a talk. She received an honors Bachelor of Science in 2022 at the University of Toronto with a double major in biology for health sciences and chemistry. Okay, already you're way ahead of me. Currently, Cindy is the first-year Master's of Science student in pharmaceutical sciences at the University of Toronto. The research she conducts in Dr. Weaver's laboratory focuses on developing novel therapeutics to target excitotoxicity in Alzheimer's disease. Cindy, please tell us about that work.
Good morning, everyone. The human brain is the most complex system in the known universe. And Alzheimer's disease, AD, is the most complex disease of the brain. From the wise words of Dr. Donald Weaver himself, Alzheimer's disease attacks the very essence of what makes you, you, and me. But many individuals lose this essence that not only affects their lives, but the lives of their family and friends. Thus, it is important that something be done about this tragedy. The Weaver Lab has dedicated years of hard work to try to find a cure for this disease. Let's explore how I take part in this. An individual is typically diagnosed with Alzheimer's disease in their mid-60s, presenting some memory loss symptoms. But what if I told you that Alzheimer's disease develops decades before symptoms present themselves? One fundamental explanation for the delay between the development of symptoms, or development of disease, my apologies, to the presentation of symptoms is neuroimmune-triggered neuroinflammation that starts 20 years before symptoms present themselves. And as you can see on the screen, you can see a depiction of how the brain looks in a healthy individual compared to an individual that has inflammation taking place. And you can see how the neurons are affected when this neuroinflammation occurs. So neurons become modified by this neuroinflammation, and these modifications manifest as either a loss of function phenotype, which is hypofunction, or hyperfunction, which is a increased function phenotype. Many attempts in disease-discovering drugs have looked at this loss of function phenotype. The research I conduct looks at the latter, which is the increased function phenotype. Therefore, the research I conduct takes an innovative approach at preventing neurons from expressing this hyperactive phenotype. Therefore, exotoxicity, or hyperexcitability, can be defined as a neuron demonstrating an aberrant amount of activity when activated by a stimulus. There's a significant amount of evidence that shows Alzheimer's disease patients exhibiting hyperexcitability in cortical and hippocampal regions of the brain, whether the AD was sporadic or genetic. There are also many studies that describe hyperexcitability in AD animal models within the frontal cortex and hippocampus regions of the brain. In one study, hyperexcitable neurons were confined to the cortex close to amyloid plaques. Another study indicated hyperexcitable neurons in the hippocampus of 80 mice. In cell models, one study confirmed late-onset disease-induced pluripotent stem cells, iPSC-derived neurons, demonstrated an increase in calcium response without stimulus compared to controls. An alternative study tested 3 different types of mutations in AD in iPSC-derived neurons and found that all mutations showed hyperexcitability. Exotoxicity can also be present in other disorders. This disorder includes epilepsy, characterized by repeating seizures. A singular change in a neuron is enough to cause a seizure. Some studies suggest that 10 to 22% of AD patients have spontaneous seizures, and other studies show the presence of seizures in up to 64% of AD patients. For this reason, one promising area for therapeutic development is to repurpose anti-epileptic drugs, AEDs. AEDs are directed towards targeting one or more membrane-bound ion channel, including membrane-bound voltage-gated sodium and calcium channels, GABA-A receptors, and inotropic glutamate receptors. The issue with this approach is that the majority of anti-epileptic drugs are sodium channel blockers that have the potential to further cognitive decline. A more recently marketed anti-epileptic drug called levetiracetam is a calcium channel blocker. Currently, a clinical trial is taking place for levetiracetam as a potential drug to treat Alzheimer's disease. Yet this drug is known to have many side effects, with notable ones including memory loss, behavioral alterations, and kappa rage characterized by severe anger and aggressiveness. Therefore, having a drug with such symptoms is not ideal. Therefore, a first principle design of a drug uniquely for Alzheimer's disease is needed. In order to do this, we need to look at GABA A receptors and NMDA receptors. So first, glutamate is an excitatory neurotransmitter in the central nervous system. Glutamate binds to either inotropic glutamate receptors, such as NMDA receptors, or metatropic glutamate receptors. NMDA receptors have a binding site for glycine or glutamate. If both glycine and glutamate bind, then the ion channel will open, allowing for a sodium or calcium ion to pass through into the neuron. GABA, on the other hand, will bind to either GABA A or G protein-coupled GABA B receptors. When GABA binds to a GABA A receptor, the ion channel opens, allowing for a chloride ion to pass through the channel into the neuron. Therefore, it is hypothesized that hyperexcitability in AD arises from the upregulation of glutamatergic neurotransmission and a downregulation of GABAergic neurotransmission. Thus, the ideal drug to treat exotoxicity in AD would pose three properties. First, we would need a drug that downregulates glutamatergic neurotransmission, upregulates GABAergic neurotransmission, and since we're looking for a drug for the central nervous system, we would need it to move through the blood-brain barrier, the BBB. The solution to this is beta-alanine. Why beta-alanine, you might ask? Well, first, beta-alanine is a neurotransmitter that is naturally found in the central nervous system. Beta-alanine has been shown to have binding sites in the hippocampus, limbic structures, and neurocortex. There are also studies that have shown the twofold effect of beta-alanine being able to bind to NMDA receptors and GABA A receptors. Additionally, and very importantly, beta-alanine can also pass through the blood-brain barrier through active transport. Lastly, beta-alanine has 5 binding sites, which include the glycine coagonist site on the NMD complex, which is strychnine insensitive, the glycine receptor site that is strychnine sensitive, a GABA A receptor, a GABA C receptor, and it's able to block the GAP protein mediated glia GABA uptake. Therefore, the goal is to make beta-alanine analogs, with the idea of designing, synthesizing, purifying, and characterizing beta-alanine analogs that are alpha, beta, and N-substituted, with the goal to demonstrate both pro-GABAergic and anti-glutamatergic activities. It is realistic to conclude that more than one drug is needed to manage AD. The research that I'm conducting takes a progressive approach at finding a solution for exotoxicity in AD. This is done by using the highly unexplored beta amino acid, beta-alanine, as a backbone to create analogs that work to decrease glutamatergic neurotransmission and increase GABAergic neurotransmission. Thank you for listening. Yeah, do you want to ask a question?
Ties in very nicely to the talk we just heard about the need for multiple approaches for Alzheimer's disease. So thank you. Good luck with your work. Okay, so it's my pleasure to introduce our third speaker this morning, Dr. Albert Jin. And Dr. Jin is the Associate Professor in Division of Neurology with cross appointments to the Department of Biomedical and Molecular Sciences, and is a known specialist in cerebrovascular disease therapy. And in addition to those clinical studies, he's also has a number of important leadership roles. He's a medical director for the Regional Stroke Network of Southeastern Ontario. He also serves on a variety of Ontario health committees, including the Stroke Leadership Council, the Stroke Evaluation and Quality Committee, and is the chair of the Provincial Stroke Unit Task Group. So a lot of important roles sitting with Dr. Jin. And we're really fortunate to have him here this morning and is going to speak to us on the growing challenge of chronic neurological diseases, successful outcomes from stroke.
Thanks for that very kind introduction, and thank you for the privilege to speak to you today about the growing burden of chronic neurological disease. I come at this from the perspective of a clinician with a background in science, which really began in Don Weaver's lab at Queen's in Kingston, Ontario, over 30 years ago. Yeah, it's been a minute, eh, Don? Yeah. And as a clinician, I'm primarily a stroke neurologist, but as anyone who has spent any time doing stroke neurology knows, the fallout from that means that you get exposed to basically the spectrum of chronic neurological disease. And the challenge is immense. It's immense enough that I really have no time for any commercial interests or disclosures. Probably would explain the quality of car I used to drive up here today. But this challenge, I mean, it's too complex to really package into a short talk like this. But for people like me, where I see this every day, it really boils down to, I think, three things that we see. The first point here, I think everyone is familiar with in the room, right? We have we're getting old very, very quickly. We have a very rapidly aging population, and this is true throughout the world. The treatments that we do have, they're, quite frankly, they're not good enough, right? I mean, there was a saying from, there was an ancient Greek philosopher when he was asked, how are you going to beat, this enemy, the Persian enemy? He says, well, more soldiers, or better soldiers, or more and better soldiers. And that really describes sort of the crux of the issue right now for us dealing with chronic neurological disease. We just don't have a lot of treatments. We need more, and we need more and better of them. And the last thing is we're dealing with, as you've heard alluded to today, the most complex object in the universe, right? And we try to tackle this in the span of 5 and 10-year funding cycles. So not surprising that, progress can be a little bit slower than we'd like. I'll go to the first point. This is really not just a clinical and scientific point, but really a social and political point as well. And graphs tell you a story, and what this tells you is that we're at the beginning of the gathering storm. Right? What we're seeing here is this exponential rise in this population that is very, very vulnerable to chronic neurological disease. We're going to see an increase in the number of people over the age of 85. It's going to triple over the next 25 years, right? And are we really ready for that? We've never seen a challenge like that in our history, and this is what we're going to face now, and we're just at the beginning of it. This is what it's going to look like in Ontario. Right? In Ontario, at the very top there, the reason why it's all skewed is because they sort of squish all the different ages above 95 together. But you're going to see a tripling of the number of people over the age of 90. You're going to see, let me see if I can get this right. Yeah, you're going to see a big boost here in the number of people who are vulnerable to chronic neurological disease as the baby boomers sort of age out. And below that, you see a population of people who are it's going to expand because of population growth, but it's not going to be enough. And we know that because of this, right? We know that in this line, the red line here, this is the people that are exiting the labor market, exiting the labor force. And the blue is the line of people who are coming in. And so what we're left with is a diminishing group of people to look after a growing number of people who are going to be burdened with neurological disease. And to make things worse is that we're not actually starting from a good place, because currently neurological disease globally, I would argue, is really the greatest health challenge of our generation. Worldwide, it is the leading cause of disability. It's a second leading cause of death. In 2016 alone, the Global Burden of Disease study demonstrated that over 276 million years were affected by loss or disability globally. There were 9 million deaths, and that was in 2016 alone. And it's not getting better. We are not winning this one, right? To put that into perspective, COVID has accounted for 30 million disability-adjusted life years and 7 million deaths over the past 3 years. Compare that to what we lose from chronic neurological disease in 1 year. It's an order of magnitude greater in the span of 1 year. This illustrates that no matter where you live in the world, you don't escape this, right? Whether you live in East Asia, Western Europe, Sub-Saharan Africa, neurological disease affects you everywhere you are on the planet. You can see in this list here the variety of diseases, with stroke and dementia accounting for an enormous amount of death and disability, and migraine, which is, I think, sort of one of these underrated diagnoses because it does rob people of productivity and ability to live a full life. The thing is, though, is that no matter where you are on this list, whether you're at motor neuron diseases or stroke, it doesn't really matter because it's 276 million disability-adjusted life years in 1 year. So no matter where you are, there's too many of you if you're in this list here, and we don't have enough people. Spread over a lifetime, what you see is whether you're male or female, you're affected in the same way, more or less. In the early parts of your life, you're affected by migraine, right? Migraine, I mean, the World Health Organization considers a migraine attack to be one of the 4th most disabling things that you can have acutely because you can't do anything when you're really affected by a migraine. Ask any neurologist in the room. Something like 80% of neurologists self-diagnose themselves with migraine. My wife says that I'm not really sick, I'm just weak, but but it's a thing, so there, right? And then when you move on across the lifespan, what you see is stroke and dementia starting to take over in terms of the amount of suffering and misery this causes. You know, data tells a story, right? It's not just numbers on a page, it's not just bars on a page. It tells you a story. And what this tells you is that across the lifespan, there's this pageant of misery that starts to emerge as we get older. And we don't really have any way to deal with it. The other thing that's really worthwhile to note here is that when you can tease apart death versus disability, you know, we think of stroke, for example, as something that happens in the emergency room, hence the name, right? It happens very, very quickly. Everything, everywhere, all at once goes wrong, right? But we don't think about what's left behind, what remains, right? After you're out of the emergency room, the acute care hospital, the rehab hospital, and back in the community, this is what's left behind. And you'll notice here that the blue bars that are dark blue are almost as big as the blue bars that are light blue. You're left with the fallout from that. And that is an enormous price that our society has to pay. And so what we're left with is dealing with, you know, this wonderful thing on your shoulders, which is, you know, 7 trillion synaptic connections with thousands of processes supporting each connection. But it's also the most vulnerable, I would argue, right? That complexity accrues with it a lot of vulnerability. Over 600 neurological disease entities listed in the US National Library of Medicine. I would argue that's an undercount. There's far more than that, really, when you look at all the different ways that a disease can present. And we are still a little bit hamstrung by some of the social and political factors that are out there, right? We can't really focus in the way, or not many places in the world, I should say, can focus in the way that needs to be done to deal with this disease. This manifests throughout all levels of inquiry, whether you're in a lab doing basic science, whether you're trying to translate this into something that's relevant for people, or whether you're trying to actually test this theory in the real world in clinical trial. Stroke and dementia are 2 really good examples of that, and I'm going to use those as examples here, but it could easily apply to Parkinson's disease, ALS, et cetera, et cetera. Now, you know, we think of stroke as an acute condition, and it is. You know, there's something like 1 stroke every 10 minutes in Canada. There's 12 million new strokes every year across the world, but there's 110 million people who are living with stroke right now. That's an order of magnitude more, 10 to 1. So stroke is not just a hyperacute disease. People have to live with it. In this room, for those over the age of 25, 1 out of 4 of you are going to have firsthand experience of a stroke in your lifetime. So this is something that I would argue we're not terribly well prepared to deal with right now because, you know, I mean, we don't have a system which can really accommodate that. And we're just at the beginning of that growth curve of that population that's going to be prone to this. Now, we've had a lot of progress over the past 20 years. Treatment and prevention strategies have evolved dramatically in the past 20 years. So here's a good example of that. This is in my hospital. This is endovascular thrombectomy. It's a remarkable process, not because of just the outcome that you can sometimes affect, but because of the difficulty in organizing this process. It's like trying to herd cats, trying to get doctors who kind of really, frankly, don't like each other, trying to work together in a very short, concentrated period of time. When you think about the number of people involved for this patient, it's about 25 people that are involved in less than 30 minutes, in my hospital, less than 30 minutes, because we work fast. And that's very difficult to do. So we're proud of that. But it's not enough, because we can't help more than 10% of all the ischemic stroke patients which come into the hospital. That's the limit of what we can expect this therapy to reach. So we see this in other things. I'm the chair of the Stroke Unit Task Group in Ontario. Less than 60% of all stroke patients are getting the appropriate care they need in a therapy which we've known has worked for 20 years. Right? That's thousands of patients a year basically consigned to hallway medicine. So our current approaches, it's not enough, it's not really working well, and we have to go towards another perspective that will affect everybody at a fundamental level. We have to be able to help everybody. So I'll give as an example here a story that I think everyone in the room is familiar with, right? So this is the story of the PSD95 inhibitors, which culminated in this drug Neuroscience, which has gone to clinical trial a couple of times, and the investigation for that is still going on at the translational level. The important thing about that is not so much the drug or the mechanism. Those are all important things. The important thing about that is that this is a 30-year journey that Dr. Tymiansky had to undertake in order to get to this point, and it might still fail, right? Chances are, actually, if you're a betting person, it will still fail. Like when I said I had no commercial disclosures or interests, I really meant that. I haven't invested in any of Don Weaver's companies, right? I work very hard for my buddies, so, you know, no offense. Right? And that's just the name of the game. But endeavors like this, you can't do this in many places in the world, right? You can only do that in a few places, places which recognize the importance of basic science being translated into the clinical world and using the clinical outcome to determine where you're going to go next, and being able to sustain that sort of support over 30 plus years, right? There are very few places in the world that can do that. UHN, right, is a place that I think can do that. Krembil certainly is a place that can do that, and that's been demonstrated by the track record of so many people in this room. But despite that, it's probably still not going to be enough, right? Because 110 million people surviving stroke, and we don't have a whole lot to help them, right? 80% of these people are going to have some form of post-stroke cognitive impairment. You know, this is sort of a dementia-adjacent condition, if you will. Right? And this is true, and even if people do get back out into the community, right? Depression, anxiety, post-traumatic stress, the rates of these are enormous in stroke survivors, and equally so in the caregivers for these folks. And chronic pain is something that we see in 30%. Can you imagine living with pain? I mean, okay, so like every grown adult male remembers their fondest childhood memory is not having low back pain, but this is a different kind of pain that we see. This is the sort of pain that alters the way you live. This is the sort of pain that really changes how you view the world and, you know, what sort of hope you have for the future. Are we addressing this? So far, this hasn't been given a whole lot of attention, right? So for anyone who's interested, you know, there is a challenge for you for the 110 million global stroke survivors that are out there. And that brings us to dementia, which is an equally large global problem. 55 million people live with dementia worldwide. There's 10 million new cases a year. This will affect a quarter of all Canadians over the age of 85 with some form of dementia. And yet we're not prepared for this, right? When we look at the effect of this on caregivers, we see an enormous amount of stress, enormous amount of depression. And when you look at our primary care system, which kind of, I mean, from my perspective, kind of feels like it's evaporating very, very quickly. The ones that remain, they don't, it's probably the wrong choice of word for prepared, it's really more supported. They don't have the supports they need to take care of this growing population. We don't have the means to meet this challenge. And so we're left with this. We need treatments, we need better treatments, and we need more of them. We only have 4 that are approved by Health Canada currently. And frankly, they don't work all that well, right? We do have, you know, lecanemab, which has come online, fast-tracked by the FDA. You know, and that's welcome news because we haven't seen much progress otherwise in a long, long time. But, you know, there are questions which remain about that. And that's still a subject of controversy. You know, 0.45 point improvement on the CDR-SB. You know, is that going to change the game? I think most people would argue not. It's still welcome news. But one of the things that is sort of illustrated with that is that in order for basic science to translate well, we have to make sure we're getting to the right people. And so I was struck by this, finding from the trial for lecanemab. You'll notice here, these numbers don't add up to 100 because some people in this group self-identify as white. We look at the lecanemab trial patients here, and there's a big discrepancy here, and especially when you look at the proportion of patients over 65 who are affected by Alzheimer's disease. Something is missing. I mean, there are many reasons for this low trial participation. There's a lot of social injustice that has happened, a lot of distrust with this medical system. But despite that, and I'm not saying the trial was done badly. I mean, people did enormous work to run this trial properly. Perhaps maybe the biological status of this trial or the biological foundation of this trial could use some work, but still, it was a well-done trial, and still we have these major challenges because we're not hitting the population that we necessarily need. We need to go out there and recruit the people we need in these clinical trials to make sure our therapies are working, and we need to understand our population that we need to treat. And the only way you can really do that is to get out there and do it, is to immerse yourself in the real world. And that brings me to Don Weaver. I told Don that I would try to be restrained and tasteful. And after having spent 6+ years in his lab, that is very difficult to do. I'll put it to you this way. So there's, Winston Churchill had a comment about a famous British general, Bernard Montgomery. I think it really kind of carries over to perhaps a lot of people in this room. Bernard Montgomery, what's his famous British general, defeated Rommel in the desert. Indomitable in retreat, invincible in attack, insufferable in victory. I'll let you extrapolate from that as you will to your colleagues in the room, right? I'm not going to go over all of this, right? We know this, and people know this better than me, for example. And if you were to actually tabulate all of this, it would be the size of like an old-fashioned telephone book. If you were to recite all of this, after a while, I don't know why I keep thinking of autocratic dictators, but there's a quote from Stalin. Sorry. That was pretty cheap. There's a quote from Stalin, which, like, quantity has a quality all its own. So, we get it, Don. Yeah, I'll just go home and underachieve some more today. But what we know, what you might not know, is how it all started for Don. So, Revenge is a dish best served cold. So, as you can see here, note the smiling young physician, right, face full of optimism, right, anticipating that future of academic success and the accolades that will accrue, right, being admired for clinical expertise and being loved by friends and family alike. And of course, I'm referring to Dr. Beverly Orser, chief of anesthesia at Sunnybrook. And beside her, we have Don, still very firmly entrenched in his "don't let the barber harsh my flow" phase. He's grown out of that, as we can see. I'm not sure if that was willing or not, but he's grown out of that. And we can see that he's still hung on to that "I dress in the style of the last cool year of my life" phase. He hasn't really grown out of that. But that's Don, back in class of '81 at Queen's, and that's sort of where it started for him. He went on to finish medical school and then undertook a very, very harsh scientific training during his PhD under someone who shall remain nameless. It's kind of like Voldemort. So, yeah. Nah. And if that doesn't get you in touch with your humanity, I don't know. I mean, let me put it to you this way. This person was mean enough and cruel enough and petty enough that it makes Donald Trump look like Mr. Rogers. Nope, sorry. So that leaves Don at this point here with his first faculty appointment at Queen's University at Kingston. And this was Don's house. right? Don was the only doctor in the house, really, for epilepsy and dementia, over 20,000 square kilometers. That's the size of Israel, right? And Don was the only doctor for those people. He would see hundreds and hundreds of patients a year while running a lab. And I always found that, you know, at the time I didn't appreciate it. I said, yeah, okay, I could probably do that. I've seen it on TV. Yeah, now that I've had a chance to like walk the walk, no, I would be reduced to a weeping alcoholic with no bladder control if I had to do this. So this was where Don lived. I mean, let's put this into perspective. This is Toronto. This is what Don looked after while he was in Kingston. Yeah. And this is one of the places Don would drive to. This is the now defunct Rideau Regional Center in Smith Falls, Ontario. Smith Falls is now known for weed, right? A lot of cannabis there. Before that, chocolate. And it was also known for this place here. And this is one of the sad places in our society. This is a place for the most marginalized, the most disabled people, the people that at that time society couldn't find a place for. And so these people were kind of warehoused here, and they were kind of forgotten. And it took an hour through very poorly maintained country roads to get to Smith Falls from Kingston. And you can imagine driving that in the wintertime in a really crappy car that I recall you had. Yeah. Anyway, so Don would go there, right? And he would tell us about these patients that he would see who don't remember him. They don't know what he's doing. They don't know that he's actually trying to do the impossible for them, because these are the folks who have refractory epilepsy. These are the folks who have such tremendous injury to the brain that they will never recover. But someone has to look after them. And he went to where they lived. He met them where they're at. And I always used to wonder about that. I mean, at the time I was a chemistry grad student, and I really didn't know much more. Actually, I still don't know much about chemistry, but I always thought, like, why aren't you helping me? You know, Don, you know, I'm paid a slave's wage, and why aren't you helping me? Well, he was going to places where he could see the connection between what he does in the lab and what he needs to do in the real world. And you don't know that until you go into the real world and immerse yourself in it, and you meet people where they're at. You don't understand them until you listen to them and you go to see them. And so that's what Don did for years and years and years. This is a long forgotten document that no one has read. It's a history of the Department of Chemistry at Queen's University in 2002. And this was a quote from Don at the time. And I'm going to embarrass myself trying to say this because it's been so damn long, but you know, this is a quote from Don. I treated him intravenously with 2 milligrams of 7-chloro-5-2-chlorophenyl-1,3-hydroxy-2H-1,4-benzodiazepine-2-one and 1,300 milligrams of 5,5-diphenyl-2,4-imidazolidine dione, right? That's actually, so I'm just going to put this, that's actually how Don thinks. Okay? We all call that Ativan and phenytoin. No, Don doesn't see it that way. We see like a little bottle of Ativan, a little bottle of phenytoin, go, yeah, that's what's going to fix him. Don sees this structure. These structures are floating around in his head. And that's the connection that he makes between his laboratory and what he needs to do in the emergency room for probably some dude who was, you know, had a few too many drinks on the weekend. And yet that person requires this level of care and this level of thought behind it. So this last statement here, the future of medical therapy lies at the level of the molecule. This is not bombast. This is something which has emerged from seeing thousands and thousands of people when you were the only doctor in the house. And being one of, for that condition, over, you know, an area the size of Israel, it's not easy. And that alters the way you think, and that alters the way you do the work in the lab. And I love this quote. This was in response to an ignorant second-year organic chemistry student. I was there I was rather shocked at this, but it was a different age, and so Don kept his job. Yeah, but he's absolutely correct, right? He's absolutely correct. To the point where that idea of having to meet the challenge of this disease at all levels drove Don to go fairly deep into the weeds. So what we see here, this is a publication from way back, where it wasn't sufficient to just understand the complexity of the brain. The sort of pursuit of knowledge in this case led him to understand the molecules which are floating around in that incredibly complex object, the brain. And so some of his work actually delved into the nature of matter itself at the subatomic level. I mean, how many clinical neurologists do you know are going to talk at length and write a paper using semi-empirical molecular orbital theory and ab initio molecular orbital methods to understand how these molecules work, these molecules which can save us or kill us, right? That's really where the focus was. It wasn't on understanding, you know, the beauty of nature and that sort of thing, although that's certainly there. The idea here was to understand these things can kill us. These things can save us, and I need to understand that if we're going to meet the challenge of neurological disease in the future. And that's one of the things which drove Don. And that leads into the next step, which is to translate this into the real world, to find animal models where you can actually test these theories and then to use those findings to go into the real world. And this is all about playing the long game. Like I said, I haven't invested in any of Don's companies, and frankly, I never had enough money to do that anyway. But you know, Don has recognized that a lot of ideas they wither on the vine, right? There's this sort of number that's thrown about, 17 years. 17 years to take a new idea from the moment you have it to the moment that it's actually translated into the real world. Along that way, about 85% of the literature behind that idea is attrited. It's lost. It never really reemerges within a few years after its initial publication. And as a result, we have companies that eventually emerge, and you need these companies because otherwise that attrition is for nothing, right? Those ideas will disappear. And so you have to push this to a level where you have to try to commercialize, and you have to try to get this out into the real world. Now this was trimiprosate, which is, I think, homotaurine? Homotaurine, yeah. 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 And it's one of these things which had a lot of theory invested into it. And until you go to clinical trial, this is what you have, right? Yeah. This was actually on the acknowledgement page of my PhD thesis. Yeah. See where that got me. Anyway, so the need is to commercialize and to get out into the world and to test these ideas in the real world, because the brain is too complex for us. It defies all of our expectations in health and disease, and our theories will fail, and we won't know they fail until we actually test them in the cold light or truth of a clinical trial or through a clinical study. So the research here tells us what we need to do, where we fail, where we need to go back to the drawing board again. Clinical research is just as important as the basic science and the translational research out there. It's one of the three main pillars that's going to drive our ability to meet the growing challenge of neurological disease. And it's really, really important, because outside of places like this, it's not a very pretty sight. Outside of places like this, the growing challenge of chronic neurological disease is utterly devastating, right? So this is something I see several times a day, right? This is a quote. The first quote there is from a Japanese monk during a long and forgotten war, the Imjin War in Korea, after a particularly bloody battle. Whoever sees this, out of all the rest of their days, today has become the rest of their life. And I would say that applies to everyone, pretty much everyone I see who's had a stroke, pretty much everyone I see who I have to break the diagnosis that you have an incurable disease, you have dementia. We will do our best to look after you, but we're not going to win, or at least not today, right? That moment when this happens, every physician who's had these moments, you know, these are the days where you give a little bit more than you get. The second quote here is from Don. This is from a poem that he wrote called The Enemy of Humankind, Alzheimer's Disease, The Enemy of Humankind. These glories of our life, our race, grand triumph of our brains, time, space, diseases of our brain and mind, true enemy of humankind. And I like this poem because it really brings the effort, all the research and all those years of accomplishment back into really the sort of distilled human empathy that you really need to drive this over the course of a lifetime. we must embrace research and strive. We must caress the tragic lives of those AD afflicts, invades, protein and mind misfold pervades. To cure AD, the mind, the brain, with scientific hand humane. And this really does speak to, I hope, what drives everybody in this room. Because what you people do here is not really just a scientific activity. It's a very human activity that we desperately need, because we are not currently up to the task of facing the growing challenge of chronic neurological disease. And for so many people out there in the world is a very dark place, right? They live in perpetual twilight, and they're hoping, even the hope for a better world or for a better time is based on a fear of what they're going through right now. And so to those of you who are carrying on the fight and who are going back to the lab to find a better world for the rest of us, and including those who actually deal with this every day, you have my heartfelt thanks and heartfelt respect, and thank you very much. That's it.
Well, thank you so much, Dr. Jin. I see a little bit of resemblance, or at least transfer of something that from that time in the. So, as you heard just now, and if you were wondering about the title of this symposium, the title of the symposium was actually Molecules, Poetry, and Exploring New Approaches to Big Challenges in Neuroscience. And several people have spoken about sort of the unique aspects of Don that he's brought to this role, both his role as a neurologist and his role as a scientist, his role as a leader. But even more sort of broad or interesting than that is that Don has also had a very special and large interest in poetry. And I think it was Jaydeep that said, with science that doesn't come out of the lab and doesn't reach publication doesn't have impact. It's actually true for Don's poetry too. So Don is a published poet and has published numerous poems. And I don't really know all the reasons why Don, what drives his interest in this, but I think it's twofold. Number one is for his patients, and he's written poems to help with on the therapy side in the absence of those molecules he's going to create in the future years. And I think he's also done it a little bit for himself. And you heard about the challenge that someone has in treating the patients that you just heard about there, and not having the kinds of therapy and solutions, the medical solutions you'd like to do it. And I think the poetry itself has also helped been a balancing piece for Don, but I'm sure we'll get a chance to hear a little bit more from him. But I think it just speaks to the sort of the depth and the breadth of the unique personality and leader that Don has been. And with that, I have two very special announcements to make this morning. We've tried to keep this a little bit quiet and on the down low, but I think maybe one of them has not been so quiet. And the first of those is that last night, we had the honor of being able to present to Don a new chair in, let me just get this right, drug discovery in Alzheimer's research. And this chair does two things. Number one, it recognizes the exceptional work that Don has played over the last 10 years, both in his lab and as a leader at the Krembil. And equally important, it recognizes a new level of support to enable the research that is going to take place in Don's lab over the next 5 years as he rededicates himself and his lab and the people who he affects around him, around the cause and the design of new molecules and the opportunities to treat dementia and Alzheimer's disease. So we're extremely excited about Don's appointment to this new chair. I also want to acknowledge the Krembil Foundation and the Krembil family, who have been long supporters, as you know, of the Krembil Institute, the Krembil Brain Institute, of Don and his work. And through this new chair, we'll continue to support Dawn and the impactful science that's going to come. So a big thank you both to the Krembles and a thank you to Dawn for taking this on. Okay, and the second announcement, and Dawn, I hope this one's a bit more of a surprise for you, is the announcement of the Donald Weaver Postdoctoral Researcher Fellowship for Research in Neurodegenerative Disease. So this is a new competitive fellowship that will award and support a postdoctoral researcher annually whose research is in the field of neurodegenerative disease. It's going to be managed by the Kremble Research Institute, and the first call is going out later this week. Okay, and with that, it's my great pleasure to invite the man of the hour to the podium, Dr. Donald Weaver.
Well, I won't be inviting Al again. Anyway, thank you ever so much. Today's been absolutely wonderful. It's been very touching. And I must say that the presentations that have gone on so far have been truly spectacular and puts on a fair amount of pressure for me not to screw it up. So if you are really an immunologist, please plug your ears for the next half hour because what you're about to get is immunology from the perspective of someone who did a PhD in synthetic organic chemistry. One of a paper that we recently had out on the immunology of Alzheimer's disease, which came out in Alzheimer's and Dementia, a very reasonable journal, probably the major journal in the Alzheimer's space, went to 9 reviewers. And I remember the first review was, he's thinking so much out of the box that this paper does not belong in any journal, this one included. And I took that as a compliment. And they actually had to, they would send it out to 2 reviewers, one who said, this is despicable, and the other one went, oh my God, this is wonderful. And they sent another 2 and got it. And the editor actually was communicating with me, and he said, my goodness, it's split reviews every time, and so we're running 50-50, 50 accept as is 50, oh my God, you have to reject this. So he said, we're going for a roll of the dice, and I'm going with 1 more. That was the number 9 reviewer. So either the vote will be, 5-4 against or 5-4 for. And it came out last year. So that showed you that they actually picked a good reviewer for a change. So what I'm going to do is talk about Alzheimer's disease and trying to think of it in a different sort of way. I'm not saying this is right. I'm not saying it's wrong. I'm saying we just have to think about it differently, and hopefully this somehow triggers people to think about Alzheimer's disease in different ways, because we bloody well need that. So as we've heard, Alzheimer's disease is one of humankind's leading causes of dependency, disability, and death, and the WHO recognizes it as a global health priority. And it really is a global health problem. Currently, 52 million people in the world are living with Alzheimer's disease or related dementias, and by 2050, we'll be up to 150 million people. And it's growing. It's growing in every continent. It's growing in every country. And it's growing in areas that have developing economies. And so this is a global problem, and it needs a global solution. And as has been mentioned earlier today, some of the solutions are very expensive. And that's why we are passionate about small molecules. We're passionate about small molecules because we think that this has the capacity to have an international presence and international penetration in the treatment of this devastating disease. And yes, it's a global problem, but we have to also constantly think of it at the level of the individual. This is a bloody awful disease. It's awful for the family, it's awful for the patient, it's awful for the family, and for society as a whole. And this is costing us a tremendous amount of money. For the sake of people who are afflicted with it, and for the sake of our own economies, etc., we really do need to move ahead in our understanding and treatment of this disorder. To date, the small molecule world in the treatment of Alzheimer's disease has not been stellar. There's lots of compounds out there, donepezil, rivastigmine, galantamine, memantine, but they're symptomatic at best, and that's probably being generous. They really don't do anything to affect the natural history of the disease. They're not DMDs, they're not disease-modifying drugs. As we've heard, the world of Alzheimer's disease is a long list of failures. And this is a scheme that shows a 10-year period from 2008 to 2019, and the most telltale is the phase 3. The number of successful phase 3s in the Alzheimer's space, in that 10-year space, 0. So this is certainly a disease which has a long and troubled history, and a history that is marked by significant failure. Now, as we've also heard, there's been some change in the past year or two. There was aducanumab, last year I was called by some students at the University of Toronto to say, we have a group project on aducanumab, can we talk to you? And I said, certainly. What science program? And they said, oh, we're not in science, we're in the School of Business. And we're doing this as a case study of how not to introduce a drug. So it's attracted more study in how not to launch a drug. So aducanumab has not done particularly well. Lecanemab has come along, and it's certainly not a cure. We've heard, what does a 24% improvement in memory really mean? Probably not a hell of a lot. But if you keep people out of nursing homes longer, that is useful. So there are some uses here. I like telling people lecanemab is a step in the right direction. It's just not a big enough step, not nearly big enough. And so we really do need to really keep pushing here, keep pushing as we move forward against Alzheimer's disease. And we really do have to keep thinking about, where are we going to go in trying to come up with curative agents? We don't need any more symptomatic stuff. We need disease modifying. We need things that are really going to change the natural history of this disease. As we've also heard, how did we get here? Why are we still flailing after so many years into this? There's this amyloid hypothesis, which was described much more eloquently earlier today than I'm about to. But this is basically the idea that amyloid misfolds, and when amyloid misfolds and aggregates, bang, that's when all bad things happen. It's popular to bash the amyloid hypothesis. I don't bash the amyloid hypothesis. Ultimately, I think it's going to be a cocktail of drugs, and depending if we're smart enough to recognize the types of dementia, the types of Alzheimer's disease, it may have a role in there. It may have a role in the individually selected cocktail that people will get. So I don't think we should say the amyloid hypothesis is at the window. It's not. It's certainly an area that still deserves some study, and I think that amyloid will continue to be relevant as we go forward, but it's not the only game in town. It's only one game, and we need to have a lot more activities going on. So the big issue here is that Alzheimer's disease, especially late onset Alzheimer's disease, is bloody complicated. It's got a pathogenesis and a pathology which is just very, very challenging. There's just so many different cell types, so many different macromolecules and small molecules involved in it. it really is quite tough. And that's why we have, people going, it's a proteopathy. No, it's an immunopathy. No, it's a mitochondriopathy, And, I like going to the Alzheimer's conferences where people, stand up and passionately say, that their approach is right or whatever. But ultimately, we're going to have to hit as many of these as we can. This is going to be complicated, and this is going to be a major hurdle to therapeutics development as we move forward. So, we'd like to talk about we need to think outside the box. We need new ideas, new directions, or don't think outside the box, think like there is no box. The frustration with the amyloid hypothesis is present in many areas. How Not to Study a Disease was an entertaining book. Basically it says, boy, we put a lot of effort into the amyloid hypothesis and it just hasn't worked out. And so this has resulted in this overwhelming need for us to really think about new ideas and new directions. And so this is where I have to, stop and look in a mirror and go, do I have that capacity? Does a partially fossilized 65-year-old brain have that capacity to go in new directions? And think back on the years, when, 30 years ago, think how desperate I was to take graduate students like I did. So things have improved since then. So yeah, I have spent many years seeing patients and was mentioned over the course of the past 30, 40 years. I've had the unfortunate task of telling approximately 10,000 people that they had Alzheimer's disease and that I was probably not going to do much for them. Neurology is referred to as the diagnose and adios specialty. We see you and then we say, sorry, don't have anything for you. Geller is not here, so she can't gloat from the perspective of a neurosurgeon. Realizing this, became interested in commercialization of research early on, and had a company called Neurochem, which we did at Queen's, and I mean, we took Neurochem to an IPO. It was traded on NASDAQ, et cetera. We raised about $120 million for a phase 3 trial on our compound, which failed gloriously. And of course, in retrospect, when we looked at some of the results that we had from that trial, a lot of the patients didn't have Alzheimer's. It was a bloody mess. Although we had some nice imaging studies from that trial. And more recently, we've had Treventis. Treventis has been focusing on tau and tau pathology. And as you've heard, Treventis has had good fortune recently in partnering with Takeda as we hope to move ahead and ultimately achieve a clinical candidate. So, I've really been focused on proteins and protein misfolding, be it amyloid, which was trimiprazate, or tau, which is Treventis. But as that complicated picture showed, there's a whole lot more here that we have to be thinking about. And so let's return to first principles. Let's go back to the drawing board, and let's try to rethink Alzheimer's disease. And maybe we're right, maybe we're wrong, but we're having fun. What we've done is a multi-step study on trying to devise a new model of Alzheimer's disease. And when I present this at conferences, when I present this in submissions to journals, you get people who are supportive and people who are really quite opposed to it. That's good. That's good. I support their right to be wrong, of course we're bloody well right, and you do have to have that mindset to keep playing in this particular space, which has got just so much failure in it most of the time. So what we did is we started off with some in silico studies, so computational simulations, And you know, because of my background in computational chemistry, I like in silico studies. It's not in reality, but it's in silico, and you can get very good insights from this. So we started off with model membranes of neurons, and we just put beta amyloid on top of it, and you know, hit go and let it convulse away up there. You know, interestingly, part of the beta amyloid stays on the surface of the membrane, and part of the beta amyloid wiggles its way through the neuronal membrane and ultimately breaches the neuron, resulting in neuronal death. And this just shows the blue, correct? Sorry, I'm a little color challenged. I usually have to pre-memorize my colors. The blue beta amyloid working its way through the neuronal membrane. And so really what happens here with these in silico studies is that the end portion of beta amyloid, particularly around the HHQK tetrapeptidic region of that, electrostatically binds to the surface of the neuron, and then the C-terminal portion, which is primarily lipophilic, gradually works its way through the neuronal membrane and breaching it. And when you look at that, you go, I mean, these are calculations that we did, 20 years ago. That's just like an antimicrobial peptide. That's how they kill things. And that's how, you know, if you look a lot of chemokines and cytokines and immunopeptides, a lot of things behave in this way at the molecular level. And so, you know, we became interested then in reframing beta amyloid as an immunopeptide. And so we came to the conclusion that A beta is an innate immune system immunopeptide. We did work to verify that it was an antimicrobial peptide, and by no means are we the only individuals in this. Of course, there are multiple other labs who have also done this work, and it showed that A beta has both antibacterial and antiviral properties, and that really does very nicely confirm some of the simulations that we did. And so, you know, we can say A beta behaves as an immunopeptide. And if it's an antimicrobial peptide, then clearly it's part of the innate immune system, because antimicrobial peptides are part of innate immunity. So, you know, this was an interesting thing for us. And we've even extended this recently and asked the question, is A beta just a cytokine? Once again, it only took 5 reviewers. But last night, actually, during dinner, I got an email from Alzheimer's and Dementia that they accepted the paper, which its title is Beta Amyloid is a Cytokine. And in it, we defined the 30 fundamental features of what makes a cytokine, and show beta amyloid satisfies all 30. And so we're saying that you know, it behaves as an immunopeptide. It's definitely an antimicrobial peptide. It may even be a cytokine. It really plays a big role in the immune system. And in that way, it's not the big evil thing. It's part of our immune system. You know, it really is quite necessary. So that's nice. And if we frame it and we say beta amyloid is an immunopeptide and it's part of the innate immune system, the next question that pops up is, why is Alzheimer's a chronic disease? Because innate immunity, it tends to be acute, it tends to be fast, you know, and when we think of chronic things, like most, you know, we tend to think of adaptive immunity as being the more protracted one. And once again, I said remember, this is an organic chemist telling you immunology. How is that? How do we get the chronicity picture in here? So if you take neurons and you kill them by necrosis, or you kill them by apoptosis, and you take the death products and you sprinkle them on adjacent neurons, the necrotic ones, when you sprinkle them on the adjacent neurons, trigger that to produce more beta amyloid. The apoptotic ones, not so much. And so you go, neat. There's something from necrotically killed neurons, and by the way, when beta amyloid comes down and punctures its way through, you're killing that neuron by necrosis, that then migrates to an adjacent cell, produces more beta amyloid, triggers it to produce more beta amyloid, and that is chronicity because you just keep triggering, self-triggering. And so we actually did some work there and demonstrated what that was. And so A beta complexes with something called GM1, a ganglioside that's in the cell membrane. And the A beta GM1 complex is what actually diffuses to adjacent neurons and causes it to produce beta amyloid and go on and on and on. So here we have this insight into how chronicity may be happening here. So what I am suggesting is as follows, is that in response to various stimuli, infection, trauma, ischemia, air pollution, depression, A beta is released as an early responder immunopeptide triggering an innate immunity cascade in which A beta exhibits both immunomodulatory and antimicrobial properties, whether bacteria are present or not. And this results in a misdirected attack upon self-neurons arising from analogous surface topologies between neurons and bacteria, rendering them similarly susceptible to membrane-penetrating attack. Following this attack, necrotic neuronal breakdown products diffuse to adjacent neurons, eliciting the further release of A beta, leading to a chronic self-perpetuating autoimmune cycle. AED thus emerges as a brain-centric autoimmune disorder of innate immunity. And normally when I get beat up by the referees, what they hate is autoimmune disorder of innate immunity because they always tell you autoimmunity is adaptive immunity. Didn't he read the introductory textbooks? And I like to tell them my mind wasn't troubled by them. So that I was actually able to think. So if we put this in a somewhat more of a pictorial view here, up at the top in the red, infection, trauma, ischemia, whatever, really does result in the release of beta amyloid as an immunopeptide. And we're going through two arms here, an autoimmune arm and an autoinflammatory arm. But the autoimmune one is the one that we're focusing on, where it causes necrosis of neurons, and then the A beta GM1 complex goes back and loops this into a cycle. But I mean, you know, you can't ignore the other half of the slide, the purple half, where we have a whole lot of other immune properties going on here. And so this really is an extremely complicated entity, which you know, everyone in this room appreciates. Now, the big interesting part here, and a central part of this notion, is that A beta can't differentiate bacteria from neurons. And so, because we're saying that A beta is released as an immune peptide, you know, and it's an antimicrobial peptide, so if there's bacteria out there, it's happy to kill them. But if when it's on its search and destroy mission, if there's no bacteria around, it looks at neurons and says, you'll do, you're a good target, and starts binding to them and killing them. And you kind of go, and we've done the molecular dynamic simulations of putting beta amyloid on top of neurons and putting beta amyloid on top of bacteria. It kills them the exact same way. And you go, does that make sense? Should bacteria and neurons be equally susceptible to this? And you know, like, why is this happening? Well, you know, it's happening because the antimicrobial peptide really can't differentiate bacteria from neurons. And if we do an electrotopological map of neuronal and bacterial membranes, looking at where there's charge, looking at where there's zones of lipophilicity, we can actually overlap, especially at the synaptic area, overlap. Bacteria have a transmembrane potential gradient, much like neurons. You know, there's a whole lot of similarities here. And so really when it comes down to it, you know, we appreciate that bacteria within a biofilm communicate with each other very similarly to neurons. And so this really sets us up for having our brain susceptible to an antimicrobial peptide, which is beta amyloid, and that this triggers a whole immune mess, which has so many implications across so many systems in the brain. So with that in mind, what we then said was, you know, if it's something to do with immune, Immunity is one of the most highly regulated systems in the body. I mean, it's crucial to our homeostasis. And therefore, there should be some endogenous factors out there within the brain which would help modulate this. And so using infinite discrimination, we took about 1,200 small molecules from the brain and screened them all. And we basically screened a large, 1,200 is not large, but reasonable sized library of small molecules endogenously occurring within the brain and looked at them for their abilities to block protein aggregation, but more importantly, for them to work against the ability of, say, of microglia cells to produce pro-inflammatory cytokines, IL-1 beta, IL-6, TNF-alpha, etc. And what we found in screening all these endogenous compounds is that there are two metabolic pathways which emerge as absolutely crucial. One's tryptophan and one's arginine. Tryptophan more so. And so that there's a wide range of metabolites of tryptophan which really do have the ability to downregulate the immune system within the brain and thus to potentially act as a therapeutic. And I draw a parallel here to the age-old use of steroids to suppress immune disease. So if somebody comes in with a severe flare of their lupus or whatever, it's not unusual for them to receive steroids. Steroids is an endogenous regulator of adaptive immunity. We are proposing that amino acid metabolism could be an endogenous regulator of innate immunity. And this isn't exactly a revolutionary concept. Oncology has been looking at tryptophan metabolism and its ability to affect tumor immunology for years. So, there's background here across the disciplines to justify this particular approach. And so, one of our academic projects that we have ongoing is how can we use this identification of endogenous, I'll call them anti-Alzheimer's or anti-innate immunity molecules, to try to come up with small molecules which may have efficacy in suppressing the immunopathology of Alzheimer's disease and may be complementary to the other approaches. Maybe, in this cocktail that I'm talking about, maybe we need a small molecule immunoactive agent. And so we have certainly been looking at tryptophan metabolism. One enzyme in particular, IDO, which is the first step in the enzymatic catabolic breakdown of tryptophan, has been of interest to us. And so we have been working on coming up with IDO inhibitors. And once again, this isn't unique. The oncology space has had work on IDO inhibitors, but for them, crossing the blood-brain barrier is not important. For us, it's everything. And so here's one where we couldn't really go and repurpose a drug from oncology. They have some phenomenal IDO inhibitors, but they just have no bioavailability to the central nervous system. And so, as usual, as we do in so many of our efforts in our lab, we start off with the enzyme. We do molecular modeling of the enzyme, try to come up with small molecules that will fit in it, and start making analogs. I call this the methyl ethyl propyl futile approach to med chem, but it works. You make lots of analogs, and when you get the one that works, in retrospect, you say, of course it worked, and I knew it all along. You give it back to the molecular modelers who tweak their model and demonstrates how it fits perfectly, and this was obvious from the beginning. So, this is ongoing work for us, and we certainly are using multiple approaches, not just IDO. We have multiple ongoing approaches right now at trying to come up with small molecules which will certainly influence the immune system, the innate immune system within the central nervous system. But as we've heard repeatedly today, the road to therapies, it's going to be a difficult road. This is one of the roads that will be replaced in the Toronto City upgrades coming up. That was the road that Doug Ford rolled on. Oops, sorry. So we need an agent which is efficacious, safe, brain penetrant, widely available, but we have to appreciate that Alzheimer's is more than one disease. It's a syndrome, it's not a disease, and we're going to have to come up with the need for combination therapy. You've seen some of these pictures before, and I'll show them again. I always tell everyone that I'm just the eye candy for our lab, and that was not funny. That was that was a statement of fact. There are other people who actually do the real work, so I owe a lot of thanks. First and foremost, I will thank my wife. It takes a whole lot of patience and tolerance to live with me. And yes, I did meet her when she was 3. And no, I wasn't 20. I was 5, okay? So yes, we have been together that long. God, she's lucky. I'd also like to acknowledge the many, many people who have worked in my lab over the years, the many graduate students, postdocs, technicians. There are many there, and colleagues that I've worked with in the chemistry setting of our lab. In particular, I'd like to acknowledge my 2 colleagues, Chris Barden and Mark Reed, who I've worked with for more than 20 years, and whose expertise and advice on a wide range of issues is always crucial to the success of our work as we move forward. And so it is a tribute to them. And of course, since head trauma is a risk factor for Alzheimer's disease, there's nothing more important than putting hockey sticks in their hands at least once a year and seeing what happens. And it's nice because I am the boss, the PI of the lab, and older than everyone else. And so I hit people maybe when we're playing hockey, and I notice that they don't hit back, which is, damn, it's a nice position to be in. Although a couple of years ago, one of the grad students didn't know who did it. Oh, man, he rattled me. Oh, sorry, Dawn. Anyway, drug discovery is a team sport, and I've had the best of luck in having a wonderful team over the years. We also have a wonderful... we also, in my past 10 years as director here, have had the wonderful opportunity to work with my many colleagues across Krembil and UHN, and I've also had the absolute delight to work with an amazing administrative team. They have made the 10 years as director happen in a good way. And once again, I would just like to acknowledge the important duo. That important duo being Carly McPherson and Amy Ma, shown here. Whenever we ran into rough waters, which was not infrequent as we tried to move the Krembil Research Institute ahead, it was their skill which let us get through these stormy times and these rough waters and emerge safely on the other side. And so to Carly and Amy, I give my utmost thanks and say that I couldn't have been director without you. As also we've heard, this costs a lot of money. Doing the methyl ethyl propyl futyl approach, that burns a lot of money. I've been fortunate to have had an awful lot of support from many, many different sources over the years. And this lists support over the course of the past 20 years, some of the various funding groups that have funded us. And at top and up center is the Kremble Foundation, which has been an immense supporter not only of the Kremble Research Institute, but also of my lab. And their dedication to Alzheimer's disease and dementia is deeply appreciated, and their contributions to the success of this institute and of my lab is amazingly appreciated. So thank you ever so much. And I'm just going to bring it full circle with my final slide, you know, try to frame this on you know, you fail all the time in Alzheimer's, and you know, how do you get up every morning and come in knowing that you're going to fail again? You know, what are the motivating factors here? And as you heard mentioned earlier, over the course of the past 40 years, I've seen 10,000 people, you know, diagnosed 10,000 people with either Alzheimer's or some related dementia. And I don't have anything for any of those 10,000 people. That's not nice. You know, damn it, we have to do something for them. You know, is our lab going to do it? I don't know. I hope so, but I hope we at least have contributed to the knowledge base which enables it. I think often of the very first patient I saw with Alzheimer's disease. It was September 1981, and yes, I was seeing patients in September 1981. You can do the math, you can go, damn, he is old. I appreciated Cindy starting off with, mid-60s is the high-risk zone for dementia. Yeah, it's okay, I'll forget. But the very first patient I saw with Alzheimer's disease was in Kingston General Hospital, because I was at Queen's at the time, I was, you know, junior house officer, and was paged to go see, you know, to come to a problem on one of the wards. So I went up the ward, and there was this 84-year-old man, and he'd crawled under his bed. He was sundowning, so that, you know, as you get with Alzheimer's disease and dementia, when the sun goes down, the degree of confusion and whatnot gets worse. And you know, the nursing staff said, he's under the bed, what are you going to do? And you know, I went in and I got down on my hands and knees and peered under the bed at him. And it was heart-wrenching. You know, this poor man was just looking at me with absolute terror in his face. He was scared. And he was, unfortunately, reliving some events from World War I. He was, you know, it was 1981, he was 84, so that puts him in his late teens when he was in the trenches of the First World War. You know, now that he was dementing, some old bad memories were coming back. And there he was, you know, cowering under and looking in absolute terror. And, you know, at that moment, you know, I looked at this poor fellow and went, God damn it, I got to do something. You know, this is pretty bloody awful. Over the years, the last 40 years since that's happened, I would say once or twice a year, I actually have a nightmare that he's in. And I still see him. I still see his face, you know, this poor man going through hell and suffering. And I have a little pet dream. It's not a nightmare, it's a pet dream, that one day I'll see his face and it has a smile on it. And it has a smile on it because we've cured this disease. And hopefully, we'll get there. I thank the Kremble family for their support of this chair. I thank Chris Page for hiring me a number of years ago, maybe against his better judgment. And I thank UHN for giving me the opportunity to work in this wonderful environment. And I can't promise you that we're going to get a cure in the next 5 years, but God damn it, we're going to try. Thank you.
Well, thank you, Don. Truly a remarkable presentation and very, very inspirational. I want to thank all of the speakers today and for everyone who's attended. You know, we've heard about the importance of research in this area. We've heard about the potential of how, when we collaborate, basic science, clinical science, back and forth, and how we can learn. We've seen spectacular failure and what that looks like. We've also seen some elements of spectacular success and what that looks like. And it reminds us of what we're trying to achieve and what we will make progress in and will make achievements in the future. So all of the talks have been very inspirational in that regard. I especially want to thank our speakers who've traveled to be with us today and to participate. I want to thank all of the people in attendance. And I also want to say a special thanks to the incredible administrative team that we have here. Don, you mentioned Carly McPherson and Amy Ma have been instrumental in helping organize today. Also to Helen Chan and to Sasha Howell and a number of others that have put this on. Today I think has been extremely enjoyable. I hope you've all enjoyed it. I certainly have. And I just want to say thanks again to Don, and we look forward to hearing about in a few years the progress that has been made under this new chair and with your new rededication to the time and effort around this area. So thank you to everyone. I'm told there are some snacks outside. You can pick those up and take those with you. And thanks again.